champs of Examsnet, welcome to the question series of Examsnet. I hope this series of detailed explanation is helping you a lot. So today we are going to discuss NEET Odisha 2019 biology paper and we are 90 questions. So let's start the session. The first question is from the chapter Cell, the Unit of Life, and the question is, which of the following cell organelles is present in the highest number in secretory cells? Options are mitochondria, Golgi complex, endoplasmic reticulum and lysosomes. And the correct option is Golgi complex. That means option number B. Okay. So these are found in highest number in secretory cells. That means their main function is secretion of the material, right? In fact, these are the secretory organelles of the cell. Secretory organelle of the cell. And who discovered Golgi complex? It was Camillo Golgi. So it was discovered by Camillo Golgi in the year 1898. Okay. Now let's have a look at the structure of Golgi complex. So as we know that these consist of many flat disc-shaped sacs or cisternae. Okay. So it looks something like this. These are the flat disc-shaped cisternae. And these are stacked parallel to each other like this. Got it? And they consist of two types of faces, convex face or forming face. So these are cis or convex faces, also known as forming faces. And the other type of face is trans face. And this is the trans, which are concave in shape. And these are also known as maturing faces. So as we discussed that their main function is secretion of the material. What happens whenever some material that is to be packaged in the form of vesicles comes from the endoplasmic reticulum towards the cis phase of the Golgi complex, and then it moves towards the trans phase and it is released from the trans phase. Okay. Why from endoplasmic reticulum? Because endoplasmic reticulum is responsible for the formation of protein. Okay. So this endoplasmic reticulum is responsible for protein synthesis, and then after that the protein enters into the Golgi complex for packaging and secretion, right? So we now know the function of endoplasmic reticulum as well as Golgi complex. What is the function of mitochondria? It helps in the synthesis of energy in the form of ATP. Okay, so the main function is ATP synthesis. Therefore it is also known as powerhouse of the cell. And the last one is lysosomes. So it is basically the digestive part of the cell, right? It is the digestive part of the cell. So the main function is the digestion of the material. So I hope this question is clear to you. Alright, now question number 2 is also from the chapter Cell, the Unit of Life. And the question is, non-membranous nucleoplasmic structures in nucleus are the site for active synthesis of. Options are protein synthesis, mRNA, rRNA and tRNA. And the correct option is option C, that is rRNA. Okay. And what are the non-membranous nucleoplasmic structures that we are talking about? We are talking about the nucleoli. So nucleoli is the plural form. The singular form is nucleolus. So suppose this is the nucleus, and we know that a eukaryotic nucleus is well defined with a nuclear membrane. So this is the nuclear membrane, and enclosed within the nuclear membrane is the nuclear matrix that is known as nucleoplasm. Now this nucleoplasm consists of some spherical bodies that are observed as dense structures inside the nucleus, and each one of them is known as nucleolus. So as we have seen that this nucleolus is non-membranous, that means it does not contain a limiting membrane. Okay. And this is the reason that the content of this nucleolus is continuous with the content of nucleoplasm, because there is no boundary here. Okay. And another important thing about nucleolus is that it is the site for active synthesis of rRNA. Okay. So active synthesis of rRNA, that means ribosomal RNA. Now these mRNA, rRNA and tRNA, all of them together take part in the protein synthesis. Okay. So let's see what are the roles of each one of them. mRNA is known as messenger RNA, and the main role is to carry the coding sequences for protein synthesis. Okay. So carry the coding sequence for protein synthesis, and it is also known as transcript. Next is rRNA that we have just now talked about. So as the name suggests, it takes part in the formation of ribosomes. Okay, so it makes up the ribosomes, and ribosomes are the structures where protein synthesis takes place. Okay, so structure where protein synthesis takes place. And the last one is tRNA, that is transfer RNA. Okay, and as the name suggests, it transfers the amino acid to the growing chain. So it transfers amino acids to the growing polypeptide chain. Growing polypeptide chain at the ribosome. So I hope this is clear to you. Now question number 3 is again related to the chapter cell, and the question is, which of the following nucleic acids is present in an organism having 70S ribosomes only? Options are single stranded DNA with protein coat, double stranded circular naked DNA, then double stranded DNA enclosed in nuclear membrane, and the last one is double stranded circular DNA with histone proteins. So here, first of all, let's see what are the types of organisms we are talking about. So it is given that the organism has 70S ribosome. That means we are talking about the prokaryotic organisms, or we can say prokaryotes. So we know that prokaryotes have 70S ribosomes. What does S mean here? S is the Swedberg's unit. Swedberg's unit, and it stands for sedimentation coefficient. Therefore, here we are talking about the prokaryotes. And what kind of nucleic acid is found in prokaryotes? Double-stranded circular naked DNA. That means option B will be the correct answer. Okay. So what does naked DNA means? Naked DNA means that the DNA is not enclosed within the nuclear membrane. So suppose this is a prokaryotic cell. So we know that here there are no nucleus, right? So there is no nuclear membrane. So the DNA exists as double-stranded circular structure, and it is present in a region known as nucleoid. Okay, so this is naked because it does not contain nuclear membrane. Okay, whereas in case of eukaryotic organisms, the DNA lies within the nuclear membrane, okay, inside the nucleus, right? So here option B will be the correct answer. And what type of ribosome is found in case of eukaryotes? 80S ribosome. Okay, but it was not asked in the question. It was asked about the 70S ribosomes. Therefore, we have to answer keeping in mind the prokaryotic organism. Question number 4 is from the chapter Cell Cycle and Cell Division, and the question is, after meiosis 1, the resultant daughter cells have option A, same amount of DNA as in the parent cell in S phase, option B, twice the amount of DNA in comparison to haploid gamete, option C, same amount of DNA in comparison to haploid gamete, and the last option is 4 times the amount of DNA in comparison to haploid gamete. Okay, so let's check. First of all, we know that cell division is of two types. First one is mitotic division or mitosis, and the other is meiotic division or meiosis. Now this meiosis is also known as half division or reductional division. The reason is that at the end of meiosis, the amount of chromosomes is reduced to half, right? Now meiosis takes place in two parts, meiosis 1 and meiosis 2. Meiosis 1 consists of prophase 1, metaphase 1, anaphase 1 and telophase 1. Similarly, meiosis 2 consists of prophase 2, metaphase 2, then anaphase 2 and telophase 2, right? Now, let's come to the question. Here it is asked about the meiosis 1. Okay, so what happens during meiosis? First, which kind of cell undergoes meiosis? A diploid cell. Okay, so this is the diploid cell, and diploid means having two set of chromosomes, that is 2n. N means one set, it has 23 chromosomes. So 2 set means 2 into 23, that means 46 chromosomes. Okay, now before the meiosis starts, this diploid cell will undergo DNA replication during the interphase. Okay, we have read about S phase. During S phase, the DNA replication takes place. That means the amount of DNA or the amount of chromosome will be doubled. So initially it was 2n. So now it will become 2 times 2n. That means 4n. Okay. Now the meiosis will start. So this is meiosis 1. What will happen? This will divide into two cells, each having 2n. Okay, so two diploid cells will be obtained after meiosis 1. Okay, so as we can see, these have been divided into two parts. Okay, now meiosis 2 will take place. Again, the amount of chromosome will be reduced to half. So this 2n will be divided into two parts, each having n. Okay, similarly, here also we'll get n. So at the end of meiosis, we have 4 gametes having n chromosomes. That means one set of chromosomes. Okay, so these are haploid gametes. Okay, each one of them is haploid. But in the question, it is asked about meiosis 1. That means at the end of meiosis 1, we can see we have diploid. Okay, this is diploid. So in comparison to haploid, the amount of chromosome is double here, right? Therefore, option B will be the correct answer. It has twice the amount of DNA in comparison to haploid gamete. Now, question number 5 is from the chapter Biomolecules, and the question is, which of the following organic compounds is the main constituent of lecithin? Options are arachidonic acid, phospholipid, cholesterol, and phosphoprotein. And the correct option is option B, that is phospholipid. Okay, so as the name suggests, it is a type of lipid. And what are lipids? These are water insoluble compounds. That means they do not dissolve in water. Now these can be simple fatty acids. Simple fatty acids, that means they will contain a carboxyl group along with an alkyl group, right? Now this alkyl group will vary from one fatty acid to another. That means the number of carbon atoms in alkyl group will vary. For example, if we talk about the palmitic acid, so this is a lipid which is simple fatty acid, and it contains 16 carbons including the carbon of carboxyl group. Okay. Similarly, arachidonic acid, that is also mentioned in the options. So it contains 20 carbons including the carbon of carboxyl group. Okay, so total number of carbons are 20 here. Now, so these are one type of lipids, but another type of lipids are those which contain some functional groups. Okay, so some functional groups are also there. Functional groups, for example, cholesterol. This is also mentioned in the options. So it consists of a hydroxyl group, right? Hydroxyl group. Similarly, phospholipids contain phosphorus or phosphorylated compounds. And lecithin is one of the examples of phospholipids, right? So I hope this is clear to you. Next question is from the chapter Transport in Plants, and the question is the main difference between active and passive transport across cell membrane is option A, passive transport is non-selective whereas active transport is selective. Option B, passive transport requires a concentration gradient across a biological membrane whereas active transport requires energy to move solutes. Option C, passive transport is confined to anionic carrier proteins, whereas active transport is confined to cationic channel proteins. And the last option is active transport occurs more rapidly than passive transport. Okay, so the correct option is option number B, that is passive transport requires a concentration gradient, whereas active transport require energy. Okay, so let's write down the main difference here. In case of passive transport, what happens? The molecules move from higher concentration to lower concentration. Okay, higher concentration to lower concentration. That means they move along the concentration gradient. And this is known as downhill movement. Hill movement, because the movement is taking place from higher to lower concentration. Whereas if we talk about the active transport, just the opposite is true in this case. That means the molecules in this case will move from lower concentration to higher concentration. Higher concentration. This means they will move against the concentration gradient, okay, not along the concentration gradient. And this is known as uphill movement. Now, in case of passive transport, since the molecules are moving along the concentration gradient, the process will take place naturally, or the process will take place very easily. But in case of active transport, since they have to move against the concentration gradient, therefore the process will not take place easily and energy will be required. Okay, so energy is required in order to carry out the active transport. Okay, and another important thing in case of active transport is the involvement of membrane proteins. Okay, so membrane proteins, which are known as pumps, are required here. Okay, so what do these pumps do? They use the energy to carry the substances across the cell membrane, okay, from one side to another. Now these membrane proteins are very specific in what they carry. Okay, just like the enzymes are very specific in carrying out the reaction, similarly. Okay, also these are very sensitive to the inhibitors that react with protein side chain. Okay, so this was all about the main difference between passive and active transport. Now question number 7 is from the chapter Digestion and Absorption, and the question is match the items given in column 1 with those in column 2 and choose the correct option. So the first one mentioned in column 1 is renin, and what is renin? It is a proteolytic enzyme, right? A proteolytic enzyme. And where is it found? It is found in the gastric juice of infants. Gastric juice of infants means small babies, and we know that babies rely on milk. So this enzyme, that is renin, helps in the digestion of milk proteins. Digestion of milk proteins. Therefore, the correct match for A will be number 3, that is milk proteins. Okay. Now let's come to the second one. It is enterokinase. Now we know that the term entero means it is related to intestine. Okay. Therefore, we can understand that this enzyme is found in the small intestine. Okay. In fact, it is secreted by intestinal mucosa. Intestinal mucosa. And the main function of enterokinase is to convert trypsinogen into trypsin. Okay. So it converts trypsinogen, which is an inactive substance, into the active trypsin. Okay, this is active. So the correct match for B will be number 4, that is trypsinogen, because it is acting on trypsinogen. Okay, so the correct match for B is number 4. Now let's come to the third one, that is auxentic cells. Okay, auxentic cells. These cells are also known as parietal cells. Parietal cells. Now, these cells are found in our gastric glands. Okay, so these are found in gastric glands. These are one of the 3 types of cells found in our gastric glands. And what is the work of auxentic cells? It secretes hydrochloric acid. Okay, secretes HCl, that is hydrochloric acid, and intrinsic factor. Now this intrinsic factor is important here because this factor is essential for the absorption of vitamin B12. So it helps in the absorption of vitamin B12. Okay. So the correct match for C will be number 1, vitamin B12. Okay. So it actually indirectly it is related to vitamin B12 because it is secreting intrinsic factor, and that intrinsic factor is helping in absorption of vitamin B12. Okay. So the match for C will be number 1. And the last one is fructose. Now, in order to transport fructose, no energy is required. Therefore, it does not require active transport. Okay, so it requires passive transport, and it needs some transporters. So it is facilitated transport. Therefore, the correct match for D will be 2. Okay, fructose is transported via facilitated transport. So the match for D will be number 2. That means the correct option will be the last one. Option D will be the correct answer. Question number 8 is again from the chapter Digestion and Absorption, and the question is Kwashiorkor disease is due to option A, simultaneous deficiency of proteins and fats, option B, simultaneous deficiency of proteins and calories, option C, deficiency of carbohydrates, and the last option is protein deficiency not accompanied by calorie deficiency. And the last option is the correct one, that is protein deficiency, which is not accompanied by calorie deficiency. So we know that there are some diseases which are caused due to dietary deficiency of protein and total calorie intake, and these are known as protein energy malnutrition. Protein energy malnutrition, and in short it is written as PEM. Now these are caused during the times of disasters, for example drought or famine or political turmoil. Okay, and they mainly affect children and small babies. Okay, so children and infants are affected. Now there are different types of diseases. For example, sometimes only protein deficiency is there. Okay, and sometimes there is protein as well as calorie deficiency, right? So different names are given to them. First one is Kwashiorkor disease. So in this only protein deficiency is there, no calorie deficiency is there. Okay, but in case of another disease, that is marasmus, protein as well as calorie deficiency is there. Okay, protein and calorie deficiency simultaneously. So here it is asked about the Kwashiorkor disease. Therefore, the correct answer will be the last one. Only protein deficiency, not calorie deficiency. Okay, it is not accompanied by calorie deficiency. Whereas if it would have been asked about the marasmus, then what would be the correct answer? The second one. The simultaneous deficiency of proteins and calories will be marasmus, right? And what is deficiency of carbohydrates known as? It is known as hypoglycemia, right? Glycemia. And for more amount of carbohydrates than required, it will be hyperglycemia. Okay. So here, according to this question, the correct answer is the option number D. Question number 9 is from the chapter Human Reproduction, and here we have to select the correct sequence of events during the process of reproduction. Okay, so we know that gametes are the prerequisites. That means the first step should be the formation of gametes, right? So formation of gametes, and this is known as gametogenesis. Gametogenesis. Genesis means origin. Okay. So the male gamete is sperm and the female gamete is ovum. Okay. So first of all, these will be formed. Now after that, one of the gametes, that means sperm, will get transferred towards the ovum, right? So the next step will be gamete transfer. Gamete transfer. And after it reaches the ovum, what will happen? Fertilization will take place, right? So fertilization will be the next step. And fertilization is also known as syngamy. So first step is gametogenesis, then gamete transfer, then syngamy. Okay. And after that, what will be formed? A zygote will be formed. Okay. So after fertilization, zygote formation takes place. Now, after the zygote has been formed, cell division will start taking place. Okay. So, cell division, that is also known as cleavage. Then after that, cell differentiation will take place. Cell differentiation, and due to the cell differentiation, different organs will be formed. Okay. So, after cell differentiations, organs will be developed. That means organogenesis will take place. So it will lead to the formation of limbs, heart, and other organs, right? So the correct sequence should be the first one, that is gametogenesis, then gamete transfer, then syngamy or fertilization, then zygote formation, then cell division, then cell differentiation, and then organogenesis. Okay, so option number A is the correct answer. Now, question number 10 is from the chapter Chemical Coordination and Integration. And the question is, which of the following hormones is responsible for both the milk ejection reflex and the fetal ejection reflex? Options are estrogen, prolactin, oxytocin, and relaxin. And the correct answer is option C, that is oxytocin. Okay. And where is oxytocin found? It is found in the pituitary gland. Pituitary gland. And where is pituitary gland located? It is located in the brain and it is connected to the hypothalamus by a stalk. Okay, hypothalamus. Now this pituitary gland is anatomically divided into two parts. First one is adenohypophysis, adenohypophysis, and the second one is neurohypophysis. Again, adenohypophysis consists of two parts, pars distalis and pars intermedia. Pars distalis and pars intermedia. Whereas neurohypophysis contains pars nervosa. Now these different parts contain different types of hormones. Okay, so the maximum type of hormones are found in pars distalis. Let's see what are the hormones. The very common growth hormone is found here. Then prolactin hormone. Then we have thyroid stimulating hormone, follicle stimulating hormone, adenocorticotropic hormone, and luteinizing hormone. Okay, so these are the hormones that are found here, whereas pars intermedia contain only one hormone, that is melanocyte stimulating hormone. Okay, now if we talk about the neurohypophysis, that is pars nervosa, then it contains two hormones, that is oxytocin and vasopressin. Now, the important thing to note here is that these two hormones, oxytocin and vasopressin, are actually synthesized in the hypothalamus, not in the pituitary gland. Okay, but they are transported to the neurohypophysis of the pituitary gland through the neurons. Okay, and then they are stored here and then later released from here. But initially, they were synthesized in the hypothalamus. That you have to remember. So, this is the oxytocin that we are talking about in this question. So the main function of oxytocin is the contraction of smooth muscles. Contraction of smooth muscles. And we know that uterus contains smooth muscles. Therefore, during the time of delivery, the ejection of the fetus is done by the contraction of the smooth muscle of uterus, and which is responsible for the contraction of the uterine muscle, that is oxytocin. Okay. Similarly, for the ejection of milk from the mammary gland, again oxytocin is responsible. Therefore, oxytocin will be the correct answer here. Let's talk about other options as well. First is estrogen. Now, how is estrogen synthesized? It is synthesized by the growing ovarian follicles. Okay, by ovarian follicles. Therefore, its functions are related to the growth and activities of female secondary sex organs. Okay. For example, development of growing ovarian follicles, appearance of female secondary sex characters, mammary gland developments, etc. Okay. Now, if we talk about the second one, that is prolactin. Now, prolactin helps in the regulation and growth of mammary gland. Okay. Regulation and growth of mammary gland. And the last one is relaxin. So this is actually a reproductive hormone. Reproductive hormone, that means it is produced only during the time of reproductive phase. That means during the pregnancy. Okay. So this is a reproductive hormone produced only during pregnancy. And what is the function of relaxin? It basically helps in loosening and relaxing of the muscles, joints or ligaments during the pregnancy. Okay, so that it helps the body to stretch. Okay, so I hope this is clear to you. All right, now let's move to question number 11. It is from the chapter Human Reproduction, and the question is, no new follicles develop in the luteal phase of the menstrual cycle because option A, follicles do not remain in the ovary after ovulation. Then FSH levels are high in the luteal phase, LH levels are high in the luteal phase, and the last option is both FSH and LH levels are low in the luteal phase. Okay, so let's see. We know that menstrual cycle consists of three main phases. First one is menstruation. Second one is follicular phase. Follicular phase, that is also known as proliferative phase. Then this is followed by the luteal phase. Luteal phase is also known as secretory phase. And after that, the next cycle begins. Now, if we represent the number of days on this line, then we know that menstruation lasts for about 3 to 7 days. So at the end of menstruation, there will be day 7. Okay. Then day 8, 9, 10, 11, 12, 13, 14, 15, 16, and so on. Okay. Now this day 14 is crucial here because this marks the middle of the menstrual cycle. Okay, so around this time we are in the middle of the menstrual cycle. Now what happens in menstruation? This is the breakdown of the endometrium lining of the uterus. Therefore the blood flows during this time, right? Now after that follicular phase follows, and what happens during the follicular phase? At this time primary follicles in the ovary grow to become fully mature Graafian follicles. Okay, so these are the primary follicles, these are developing, and at follicular phase there is a fully mature Graafian follicle. Okay, this is Graafian follicle. Now let's represent the variation in the pituitary hormones. Okay, so first hormone is luteinizing hormone. So during until the follicular phase, the amount of luteinizing hormone is more or less same. Okay, it varies, but it varies a very little. But at the time of the middle of the menstrual cycle, that means around day 14, there is a sudden surge in the level of luteinizing hormone. Okay, this is luteinizing hormone, and this is known as LH surge. After that, the amount decreases. But here we have to see this point when there is a sudden surge in luteinizing hormone. So this surge leads to the rupture of the Graafian follicle. Okay, rupture of Graafian follicle, and due to that rupture ovum is released. Ovum is released. So this is how it is ruptured. Ovum is released from here. Therefore it is known as ovulatory phase. Okay, after that luteal phase follows. Now here the question is that in the luteal phase no new follicles develop, and the reason is, you can see that the amount of luteinizing hormone is very low in the luteal phase. Okay, it is highest around day 14. After that it decreases, so it is very low. And similar will be the variation in the follicular stimulating hormone also. Okay, suppose this is follicular stimulating hormone, so this will also vary in the similar manner. There will be surge in the level of FSH also, but it will be lesser as compared to the luteinizing hormone. Okay, so this is FSH. So we can say that in the luteal phase, the amount of FSH as well as LH are low here. Okay, therefore option D will be the correct answer, the last option. Question number 12 is from the chapter Evolution, and the question is, in Australia, marsupials and placental mammals have evolved to share many similar characteristics. This type of evolution may be referred to as, options are adaptive radiation, divergent evolution, cyclical evolution, and convergent evolution. And the correct answer is option D, that is convergent evolution. Okay. So let's see how. First we'll see the difference between convergent and divergent evolution. Now, as the name suggests, convergent means coming from different directions but converging or meeting at a common point. And divergent means starting from a common point but diverging to different directions, isn't it? Now, in convergent evolution, coming from different directions means the organisms have different ancestry. And different ancestry means the organs of these evolved species will be different anatomically. Okay, so they will have different anatomical structure, but they are evolved to perform similar function. So although they are different anatomically, they are performing similar function. So this common point shows the similar function. What can be the example? Example is wings of birds and butterflies. So although They are performing the same function, that is flying, but we should know that they have different anatomical structure. Okay. Now let's come to the divergent evolution. So it starts from a common point, that means the organism have common ancestry. So obviously they will have similar anatomical structure, but during the evolution they are evolved to perform different types of functions. For example, limbs of humans and whales. It might look surprising, but yes, they have common ancestry and therefore these are similar in anatomy. Okay, and obviously functions are different because humans use their limbs for walking, but whales do not walk, they swim in water. So the function is entirely different. Okay, now here we need to understand another important term, that is adaptive radiation. So what is adaptive radiation? It is actually related to the divergent evolution, because it is the process of evolution of different species in a given geographical area, starting from a point and literally radiating to other areas of geography, or you can say radiating to other habitats. Okay. For example, in Australia, the evolution of marsupials and placental mammals is known as adaptive radiation. Okay. Now, here we have to say that when two different adaptive radiations have taken place simultaneously, then what type of evolution it is. Okay. So this is convergent evolution. Let's see how. Suppose we have an area A, and here some marsupials are living, some placental mammals are also living. Okay. So let's write M for marsupials and P for placental mammals. Now, suppose due to some reason they are unable to live here. Okay. So what will happen? They will move to another habitat. Okay. They will radiate to another habitat, right? Suppose this is habitat B, this is habitat C. Similarly, there will be other habitats also. So in habitat B, some of the marsupials will arrive and some of the placental mammals will also arrive. Okay. Similarly, in habitat C, some marsupials will arrive, some placental mammals will arrive. Now what will happen? This area B will be different from this area C. Okay. So some of the marsupials and some of the placental mammals that have arrived to area B will be living in the similar environmental condition, or will be dependent on similar type of food resource. Okay. And the marsupials as well as placental mammals that have arrived to area C will be living in another type of common environmental condition. Okay. So obviously all the organisms that have arrived to area B, whether they are marsupials or they are placental mammals, they will be adapting to the similar condition, therefore they will develop similar characteristics. Okay. So this is a type of convergent evolution. Okay. Initially they were different, but they have arrived to similar condition, so they are developing similar characteristics. Okay. So here you have to remember that when more than one adaptive radiation takes place simultaneously due to same reason, then it can be related to convergent evolution. Okay. Therefore option D will be the correct answer. Now question number 13 is from the chapter Principles of Inheritance and Variation, and the question is match the items of column 1 with column 2. Okay, so the first one is XX and XO method of sex determination. This word syndrome should not be there. Okay, now let's talk about humans first. We know that in humans there are 23 pairs of chromosomes. Out of these 23 pairs, 22 pairs are autosomes and one pair is sex chromosome. Okay, one pair is sex chromosome. Now it varies in males and females. So if we talk about the females, they have same type of chromosome, that is chromosome X. So the pair is XX. Whereas in males, different types of chromosomes are present. One is X, one is Y. So the pair is XY. Okay. In females, it is known as homogametic condition. Homogametic. Homo means same. So since same type of gametes are present, therefore it is homogametic. Whereas in male, different type of gametes are present. So this is heterogametic. Okay. So females are homogametic and males are heterogametic. But the situation is different in different organisms. For example, if we talk about grasshoppers, here females have again XX type, but males have only one X chromosome. Okay, they don't have a pair of chromosomes, they have only one chromosome, that is chromosome X. So it is written as XO. O means nothing. Okay, so the method followed in grasshopper is XX and XO type, whereas in humans it was XX and XY. So the first one, XX and XO, will be matched with the third one, that is grasshopper. Okay, so the match for A will be number 3. That is, in option C we have this. So obviously this will be the correct option, but still let's match other things. The second one is XX and XY method of sex determination. So we have seen just now that in humans XX and XY method is followed. Okay, and here females are homogametic, males are heterogametic. So where do we find female homogametic? In option 4. The fourth one in column 2. Okay, so the correct match for B will be number 4. Again, this is the correct one. Now the third one is karyotype 45. Okay, so karyotype 45 means 45 chromosomes are there. Now we know that there are in total 46 chromosomes, but there is a syndrome known as Turner's syndrome. Turner's syndrome, this is a disorder. What happens in this? One chromosome is absent, one X chromosome is absent. Okay, one X chromosome is absent in females. Okay, so how many chromosomes left? Number of chromosomes is 46 minus 1, that is 45. Okay, and this is not a normal condition, because in normal condition we should have 46 chromosomes. So this is known as karyotype 45. 45. Therefore, the correct match for C will be the first one, that is Turner's syndrome. Okay. Now, the last one is ZWZZ method of sex determination. Now, it is followed in birds. What happens in birds? Here, in females, there is different type of chromosome. One is Z, one is W. Whereas in males, same type of chromosomes are there, Z and Z. Okay, so this is just opposite to that in humans. In humans, females have same type of chromosome, males have different type of chromosomes, but in birds, females have different types of chromosome, males have same type of chromosome. Therefore, obviously, since female have different type of chromosome, this will be known as heterogamy. So here, uh, female is heterogametic. Okay, in humans female was homogametic. So the correct match for D will be the second one, female heterogametic. Okay, so obviously option C will be the correct answer. Question number 14 is from the chapter Molecular Basis of Inheritance, and the question is, what will be the sequence of mRNA produced by the following stretch of DNA? Okay, so this is the template strand, this is the coding strand. Now, which strand is to be taken as the reference strand or the base strand? Template strand, okay, or the non-coding strand. So this is the important strand here. So let's write down the sequence of nucleotides of template strand first. Template strand, and the sequence is like this: 3 dash ATGC, then again ATGC. So here you can see we have ATGC, then ATGC, again ATGC. So 3 times ATGC and then 1 time ATG only. Okay, so ATGC, then ATGC, then one more times ATGC, and then ATG, right? And then we have 5 dash. Now, suppose first we have to copy this information into another DNA strand. That means replication is to be carried out. Okay. Then what will happen? The strand will be complementary to the template strand. That means here we'll have 5 dash and this side will have 3 dash. Okay. Now in place of A, we will write T. In place of T, we will write A. In place of G, we'll write C. And in place of C, we'll write G. Okay. So here we have ATGC, that means TACG. Then again ATGC, So again TACG, again TACG, and then TAC. Okay. Now here we have to copy this information into the mRNA. Okay. Transcription is to be carried out. Transcription. So there will be one difference only, that in place of thymine there will be uracil. Okay. In place of thiamine we'll have uracil. So in place of T we'll write U. Okay. And other things will be same. So 5 dash UACG, again UACG, UACG, and UAC. Okay. And then 3 dash. So this will be the correct answer. So let's see which option should be the correct one. Second option, right? So option B will be the correct answer. Question number 15 is from the chapter Strategies for Enhancement in Food Production, and the question is select the incorrect statement regarding inbreeding. Okay, so first let's understand what is inbreeding. Inbreeding means within the same breed. Okay, so it is actually the process of mating of more closely related individuals within the same breed for several generations. Okay, so let's write it down here. Mating of more closely related individuals within the same breed for about 4 to 6 generations. Okay, now it has several advantages. So let's see what are mentioned in the options. The first option is inbreeding helps in elimination of deleterious alleles from the population. So this is a correct statement because it helps to identify the harmful alleles that can be removed by selection. Okay, so this is a correct statement, but we have to find out the incorrect statement. So let's move to the second statement. It says inbreeding is necessary to evolve a pure line in any animal. Again, this is a correct statement because the inbreeding increases the homozygosity. Okay, increases homozygosity. Homozygosity means having identical alleles of a gene. Okay, so this way it evolves a pure line in any animal. Now let's move to the third statement. It says continued inbreeding reduces fertility and leads to inbreeding depression. So yes, again this is a correct statement because inbreeding is helpful to a certain extent. If continued inbreeding is done, then it can reduce the fertility as well as productivity, and this is known as inbreeding depression. Okay, so productivity is also reduced. Productivity is also reduced. Now let's come to the fourth statement. It says inbreeding depression cannot be overcome by outcrossing. So this is an incorrect statement because actually inbreeding depression can be overcome by outcrossing. Okay, so what is outcrossing? Outcrossing is the mating of unrelated individuals or unrelated animals of same breed. Okay, same breed, and these animals should be superior ones. Okay, so unrelated superior animals of the same breed. Now what is the advantage of outcrossing? Outcrossing helps to restore the fertility and yield. Okay, so helps to restore fertility and yield. So the incorrect statement is statement D, the last option. Now question number 16 is from the chapter Microbes in Human Welfare, and the question is a biocontrol agent to be a part of an integrated pest management should be. Options are species specific and symbiotic, free living and broad spectrum, narrow spectrum and symbiotic, and species specific and inactive on non-target organisms. Okay. So first of all, why is an integrated pest management system required? It is required for the safety of the environment, right? Safety of the environment. See, we know that today there are a lot of methods for pest control, but most of them are actually harmful to the environment. Okay, for example, if you use pesticides or insecticides, these can harm the environment and they can also affect the organisms which are actually not harmful to us. Okay, and they need not be eliminated. So we have to avoid that. Okay, for that we use biocontrol agents. Okay, in the integrated pest management system. Now, what should be the characteristics of biocontrol agent? So these agent should be narrow spectrum. Okay, these should be of narrow spectrum. That means they should not affect a wide range of organisms. Okay, they should affect only the organisms that need to be eliminated because they are harmful. Okay, so narrow spectrum, that means they will be inactive on non-target organisms. Okay, inactive on non-target organisms. Also, they should be species specific. Species specific, so that the species that is not required to be eliminated should be safe. Okay. So option D will be the correct answer. Species specific and inactive on the non-target organisms. Okay. Symbiotic is nothing to do here, and also it should not be broad spectrum. So option B can also be no, cannot be correct. Okay. So only correct option will be option D. Question number 17 is from the chapter Biotechnology, Principles and Processes, and the question is match the following enzymes with their functions. So the first enzyme mentioned is restriction endonuclease. Okay, so this is a type of restriction enzyme, and we know that restriction enzymes cut the DNA or they remove the nucleotides from the DNA. Okay, and there are two types of restriction enzymes, restriction endonuclease and restriction exonuclease. Okay, so restriction enzyme cut the DNA and remove the nucleotides. Now there are two types of restriction enzymes. First one is restriction endonuclease, and the second one is restriction exonuclease. Okay, endo means inside, exo means outside. So this restriction endonuclease will cut the DNA somewhere inside. Okay, we know that in recombinant DNA technology we have to cut a DNA at a specific location. Okay, and then insert a desired fragment of DNA. Okay, but for that first we need to cut this. So this cutting is done by restriction endonuclease. Okay, so it will cut the DNA at specific location. So the correct match for A will be the third one. Okay, cuts the DNA at specific location. This. And the second one is restriction exonuclease. So exo means outside. So this can cut the DNA only at the end. Okay. So if this is the DNA, so this end part will be removed by restriction exonuclease. Okay. So the correct match for B will be the last one, fourth one, removes nucleotides from the ends of DNA. Okay. Now, the third one is DNA ligase. So, what is DNA ligase? It acts as a glue. That means it will join the DNA fragments. Okay. Suppose this is one fragment. This is one fragment. So, these two fragments will be joined by the DNA ligase. Okay. DNA ligase acts as glue. So the correct match for C will be the first one, joins the DNA fragments. Okay. And the last one is Taq polymerase. So what is Taq polymerase? Taq polymerase is a thermostable enzyme. Thermostable enzyme. It is actually a DNA polymerase enzyme. That means it remains active even at very high temperature. Okay, that is why it is said to be thermostable, and it is extracted from which bacteria? Thermus aquaticus. Okay, so it is isolated from bacteria Thermus aquaticus. Okay, and the main role of Taq polymerase is in the polymerase chain reaction. Okay, so it helps in amplification of a DNA segment. Okay, so what does it do? It actually extends the primers on genomic DNA template. So suppose this is the DNA and this is the primer. Okay, so it will extend this primer and amplify the DNA. Okay, so the correct match for D will be the second one, extends primers on genomic DNA template. This is the last one. Okay, so the correct option will be the second one. Okay, option B will be the correct answer. Question number 18 is again from biotechnology, principles and processes. And the question is, the two antibiotic resistance gene on vector PBR322 are for, options are amplicin and tetracycline, amplicin and chloramphenicol, chloramphenicol and tetracycline and kanamycin. And the correct answer is option A, that is amplicin and tetracycline. Okay, so this is a direct question. You just have to remember this. Okay, that the two antibiotic resistance genes on vector PBR322 are for amplicin and tetracycline. Question number 19 is from the chapter Biotechnology and its applications, and the question is, exploitation of bioresources of a nation by multinational companies without authorization from the concerned country is referred to as, options are bioweapon, biopiracy, bioethics and biowar, and the correct answer is option B, that is biopiracy. Okay, so suppose there is a bioresource that belongs to a particular country. Okay, and some other country or some multinational company wants to use this bioresource, then in that case this multinational company or the country first needs to get permission from the concerned country. Okay, and then it needs to follow certain procedure. For example, a compensatory payment could be required or some other procedures could be required. Okay, but sometimes this company or this country does not follow proper procedures. Okay, it follows illegal method. Okay, and then it exploits the bioresource of another country without authorization or without taking its permission and taking all the credit to itself. Okay, so that is known as biopiracy. Question number 20 is from the chapter Organisms and Populations, and the question is, carnivorous animals, lions and leopards occupy the same niche, but lions predate mostly larger animals and leopards take smaller ones. This mechanism of competition is referred to as, options are character displacement, altruism, resource partitioning and competitive exclusion. And the correct answer is option C, that is resource partitioning. Okay, so we know that lions and leopards occupy the same niche, so they will share the similar habitat, right? So lion and leopard, suppose these two live in the same habitat. Okay, and these are the food resources available to them. Let's assume the larger circle represents the larger organisms that can be predated by them, and the smaller circles represent the smaller organisms. Okay, now if lions and leopards both target same type of organisms or both targets all type of organisms, then what will happen? This can lead to increased competition between these two. In fact, unhealthy competition between these two. So in that case, ultimately the competitively superior will be able to survive and the other one will be eliminated. Okay, so in order to coexist in the same habitat, they have evolved a new method known as resource partitioning. Okay, resource partitioning. So what happens in resource partitioning? There is partitioning of resource. That means one has decided to target the larger organisms and the other has decided to target the smaller organisms, so that this will decrease the competition between these two. Okay, and both will be able to live peacefully in the same habitat, right? So I hope this is clear to you. All right, now question number 21 is from the chapter Biodiversity and Conservation. And the question is, decline in the population of Indian native fishes due to introduction of Clarius gariepinus in river Yamuna can be categorized as, options are coextinction, habitat fragmentation, overexploitation, and alien species invasion. And the correct answer is option D, that is alien species invasion. So this is alien, not alien. Now, what is alien species invasion? Suppose we have a habitat. Now, different species will be living in this habitat, right? Sometimes intentionally or unintentionally, some alien species or foreign species are introduced into this habitat. Okay, alien species means those which do not belong to this habitat. They belong to some other habitat, but these are introduced into this habitat. So we know that this habitat is not the natural environment for this alien species. So there will be no natural predators available here, right? No natural predators. Also, there will be no similar species who can compete with these alien species. So no competition will be there. And due to this, there will be no check on their population, right? No check on population. Whereas if we talk about the indigenous species that originally belong to this habitat, they have their natural predators as well as competitors present here, right? So their population will be controlled, or there will be a balance in their population, right? Whereas there will be no control in the population of these alien species. So their population will grow rapidly, okay? Population grows rapidly. And due to this, sometimes they become invasive because the population growth is so fast that they kind of invade this area. You can say they occupy this habitat such that they pose a threat to the indigenous species living in this habitat. Okay, and this situation is known as alien species invasion. Okay, and the example mentioned in this question is of alien species invasion, because this species Clarius gariepinus, which is also known as African catfish. So it was intentionally introduced into the river. Okay, and what was the purpose? The purpose was aquaculture. Okay, so due to the introduction of this African catfish or Clarius gariepinus, there was a threat to the survival of the native catfish that lives in our river Yamuna. Okay, and this led to the situation that is known as alien species invasion. Question number 22 is from the chapter Molecular Basis of Inheritance, and the question is, match the following RNA polymerase with their transcribed products. Okay, so we know that in case of prokaryotes there is a single cell, right? So there is no well-defined nucleus in case of prokaryotes. So only one type of RNA polymerase is found in case of prokaryotes. Okay, one type of RNA polymerase enzyme. But what happens in case of eukaryotes? Here we have a well-defined nucleus. So there are different compartments here in the cell of eukaryotes. So there are different types of RNA polymerase present in the cell. Okay. So one type of RNA polymerase is found in the organelles. One type of RNA polymerase in organelles. And in addition to this one type of RNA polymerase, there are at least three type of RNA polymerase in the nucleus. Okay, so at least three types of of RNA polymerase in the nucleus. So what are these three type of RNA polymerases? First one is known as RNA polymerase 1. Then second one is RNA polymerase 2. And the last one is RNA polymerase 3. So these three are the most common ones. Now what are the products that are transcribed by these RNA polymerases? So the RNA polymerase 1 is used to transcribe rRNAs or ribosomal RNAs. Okay. So these are 28S, 18S and 5.8S rRNAs. Okay. Now RNA polymerase 2 transcribes the precursor of mRNA, that is hnRNA. hnRNA. So it is also known as heterogeneous nuclear RNA. Okay. So this is the precursor of mRNA. Now the last one, that is RNA polymerase 3. So it transcribes different type of RNAs. First one is tRNA, then 5-srRNA, and the last one is snRNAs. That means small nuclear RNAs. Okay. So now let's match them. RNA polymerase 1. So it is rRNA. So the correct match for A will be number 2. Okay. So in the third option we have A. The match for A is 2. Okay. Then the second one is RNA polymerase 2. So it is hnRNA. So the correct match for B will be number 3. Again this is correct. And the last one is RNA polymerase 3. So it will transcribe tRNA, that means C1. So option C will be the correct answer. Now question number 23 is from the chapter Principles of Inheritance and Variation. And the question is, in a marriage between male with blood group A and female with blood group B, the progeny had either blood group AB or B. What could be the possible genotype of parents? Okay. So let's check one by one. We'll draw the Punnett square for each of the given options, right? So for the first one, here we know that male has IA and small I and female has IB. Okay. So first we'll draw the box. We'll have 4 separate boxes here. Now we'll write the genotype of male on the top, IA and small I. And for female, we'll write on the left side. So it is IB and IB. You can also write the male on the left side and female on the top. Okay, according to your convenience. Now, in the first box, we'll multiply this one and this one. So IB, that means B into A. So it will be BA or AB. Okay, both are same things. In the second box, this one, B into nil. Okay, this small I shows that none of the blood groups A or B is there. So B into none, that means B only. Okay. Now in the third one, B into A, that is again AB. And in the fourth one, it will be B into none, that is B only. Okay. So we can see that in the first option only we have got the blood groups as AB or B only. Okay. So in the question also it is asked that the progeny has blood group AB or B only. So this option is itself the correct option. Okay. But still we'll check for other options as well. So for example, I'm drawing the Punnett square for second option. So on the top we'll write the genotype of one of the parents. Let's write for male. So IA. Okay. IA. This is for second option, right? Now on the left side for female, IB. Now let's write the blood groups. First one will be B into A, that is AB. Second one, B into A, again AB. Third one, B into A, again AB. And the fourth one also B into A, that is AB. So in this option we have got only one type of blood group, that is AB, and in the question it is mentioned that it has either AB or B. So there is no B here. Okay, no B is there, so this cannot be the correct answer. Now let's come to the third one. First we'll draw the square. Now the male has IA, and the female has IB and small I. Okay, just correct it. This is IB and small I. IB and small I. Now B into A is BA or AB. Now B into A again. AB. Now small I into A, that means A only. Then small I into A only. So here it is either AB or A, not B. So this will also not be the correct answer. Okay. Now we'll come to the last option here. Again, let's draw the box here. So male is IA and small I. IA, small I. Female is IB and small I. By B will be on the top. So B and small I. Now B into A, AB. B into nil, that is B only. Then here I into A, that is A, and then I into I, that is O. Okay, so here different type of blood groups are there, A, B, A and O. All type of blood groups are there. But in the question it is mentioned only AB or B. So according to the question, the correct option can be only the first one. Okay, so option A will be the correct answer. Question number 24 is from the chapter Evolution, and the question is, a population of a species invades a new area. Which of the following condition will lead to adaptive radiation? So first of all, let's understand what is adaptive radiation. We know that in case of normal evolution, one type of species evolves into another type of species. Okay, so this is the case of normal evolution. So after this evolution, only one type of new species is formed, right? But adaptive radiation is a special type of evolution, or specifically it is a type of divergent evolution. Okay, so in this, not only one type but different type of new species are formed. Okay, so let's write it down here. In case of adaptive radiation, initially we have one species, and this species belongs to a given geographical area. Okay, given geographical area, and then it evolves into different species, not just one type of species. And how does it happen? Starting from a point, starting from a point and radiating to or spreading to different areas. Okay. Now, this means that suppose initially we have a species S1. So after radiating to different areas, it will develop into different type of species, say S2, S3, S4, S5, S6. Okay. That means all different groups of these species S1 will move to different areas. They will face different type of natural conditions there. Okay. And according to that condition, they will develop into different type of species. Okay. Now, let's come back to the question. It was that a population of species invades a new area, right? Now, which of the condition will lead to adaptive radiation? So, let's see what will be the suitable condition. So, the first option is area with large number of habitats having very low food supply. So, okay, large number of habitats is okay, because more the number of habitats, more will be the environmental condition that will lead to the development of different types of species. Okay, but low food supply cannot be a suitable condition for the growth of any species. So this cannot be the correct answer. Now, second option is that area with single type of vacant habitat. Okay, so this is also not correct, because if one species will move to only one type of new habitat, then how will different type of species develop? Okay, so this is also incorrect. Third one is area with many types of vacant habitats. So yes, this will be the correct answer, because more the number of vacant habitats, more will be the type of species that will develop over the period of time. Okay, so option C will be the correct answer here. Question number 25 is from the chapter Chemical Coordination and Integration, and the question is, identify A, B and C in the diagrammatic representation of the mechanism of hormone action. Okay, so this figure is taken from the NCERT. So if you remember this figure, you can easily answer this. Okay, the diagram represents the action of a peptide hormone or protein hormone. Okay, so what is the mechanism of the action of hormone? First, a hormone binds to specific protein known as receptor. Okay, so hormone will bind to a specific protein known as receptor, and then it will form the hormone receptor complex. Okay, hormone receptor complex. Now, there are different types of receptors. Okay, some receptors are present on the cell membrane, and these are known as membrane bound receptors. So, membrane bound receptors. Some receptors are present inside the cell. These are known as intracellular receptors. Okay. Then some receptors are present inside the nucleus. So these are known as nuclear receptors. So depending on the location of the receptors, these are named. Okay. Now, what happens when the hormone binds to a membrane bound receptor? Then in that case, the hormone cannot enter into the cell. Okay. And then it has to generate a second messenger. Okay. And that second messenger will lead to different types of responses. Okay. So let's come to the diagram here. We can see that there is a molecule A. It is coming towards the molecule B. Okay. So this A is nothing but the hormone. And which type of hormone? A protein hormone or a peptide hormone. Okay. And this B that is located on the cell membrane, this is the cell membrane of ovary. Okay. Ovarian cell membrane. Now, this B receptor is present on the cell membrane. That is, it is a membrane bound receptor. Okay. So this A is binding to B. Okay. So B is what? Receptor. So in the given options, A will be protein hormone. That means either option B or D can be the correct answer. B is a receptor. So in option B, we have B receptor. In option D, it is cyclic MP. So this is not true. So in option B, A is protein hormone, B is receptor. So both are correct. And C is the second messenger that is generated by them. So this is cyclic AMP or calcium ion. So in this option, cyclic AMP is given. So it will be the correct one. Okay. So option B will be the correct answer. Question number 26 is from the chapter Human Health and Disease, and the question is, humans have acquired immune system that produces antibodies to neutralize pathogens. Still, innate immune system is present at the time of birth because option A, it is very specific and uses different macrophages. Option B, it produces memory cells for mounting fast secondary response. Option C, it has natural killer cells which can phagocytose and destroy microbes. And the last option is it provides passive immunity. Okay, so the correct answer is option C, that it has natural killer cells which can phagocytose and destroy the microbes. Okay, now we know that there are two types of immunities. First one is innate immunity that is present from the time of birth. Okay, and the other is acquired immunity that is acquired during the lifetime of an individual. Okay, so innate immunity is an example of non-specific immunity. Non-specific, and it is present in all the individuals from the time of birth. Okay, it basically consists of different types of different barriers in the body. Let's see what are different types of barriers. First one is the physical barrier. So the example is, the most important example is the skin. So this is present all over the body and it prevents the entry of the microorganisms into the body. Okay. Second example can be the mucus coating of the epithelium lining of respiratory, gastrointestinal and urogenital tract. Okay, so these are of sticky nature. So once the microorganisms enter the body, these trap the microorganisms so that they cannot spread inside the body. Okay, so they trap the microbes. Now, second type of barrier is physiological barrier. Physiological barrier. Now, what are the examples of physiological barriers? One of the examples is acid present in the stomach. Then we have saliva in the mouth. then tears in the eyes. All are example of physiological barriers. Okay. Then third type of barrier is cellular barrier. Cellular barrier, that means these are present inside the cell. Inside the cells. So when we say cell, there are different types of cells. For example, blood cell. Then we have tissue cells, cells of the tissues. The examples of cellular barriers that are present in the blood cells can be white blood cell, or we can say leukocytes, because white blood cells are types of leukocytes, right? Example can be PMNL, that is a neutrophil, and monocytes. Then in tissues, the example can be macrophages, okay, which phagocytose the microorganisms, okay. So macrophages. Now, three type of barriers we have seen, right? Fourth one is the cytokine barriers. So what happens, the cells that are infected by virus, they secrete proteins known as interferons, okay. So virus infected cells produce interferons, and these protect the non-infected cells from further viral infection, okay. So they protect non-infected cells. So we can see that this innate immune system consists of natural killer cells, and they can phagocytose and destroy the microbes. Therefore, option C will be the correct answer. Okay. Question number 27 is from the chapter Neural Control and Coordination, and the question is, which of the following statements is not correct? So we have to find out the incorrect statement, right? Now, the first statement says an action potential in an action does not move backward because the segment behind is in a refractory phase. So this is a correct statement, but we have to find out the incorrect statement. Okay. So let's come to the second statement. It says depolarization of hair cells of cochlea results in the opening of mechanically gated potassium ion channels. So again, this is a correct statement. Now, the third statement says rods are very sensitive and contribute to daylight vision. Now, this is an incorrect statement. So, let's see how is this statement incorrect. What are rods? Rods are a type of photoreceptor cells. Okay. So, we know that the retina in our eyes contain three layers. Okay. So, the first layer or the outermost layer is known as photoreceptors. Photoreceptors. Okay. So this is the outermost. The second layer of cells is bipolar cells. Okay. Bipolar cells. Whereas, so this is the middle one, right? Now the third layer or the innermost layer is known as ganglion. Again, if we talk about photoreceptors, these are of two types, rods and cones. So these rod cells are responsible for twilight vision or scotopic vision. Okay. So twilight is also known as scotopic vision. Whereas cones are responsible for daylight vision, and daylight is also known as photopic vision. So rods are required in case of dim light or night time, okay, whereas cones are required in the daytime or bright light, okay. So here in the question it is mentioned that rods contribute to daylight vision, which is not true because cones contribute to daylight vision, right? So I hope that you understood how this statement is incorrect. So let's continue with rods and cones. Rods contain rhodopsin. Rhodopsin. Again, this rhodopsin contains retinal, that is a derivative of vitamin A. And these cones are again of three types. Basically, they correspond to three different colors: red, green, and blue. Okay. And when all these three colors are stimulated equally, then it produces white color. Okay. So I hope you understood this complete thing. Question number 28 is from the chapter Locomotion and Movement, and here we have to match the joints with the bones involved. Okay, so the joints mentioned here are gliding joint, hinge joint, pivot joint, and saddle joint, and all these joints are examples of synovial joints. Synovial joints. So these type of joints allow considerable movement, therefore these are helpful in locomotion. So let's start with the first one. First one is gliding joint, and gliding joint is found between the carpals. So the correct match for one will be number 3. Second one is hinge joint. Hinge joint is also known as knee joint or elbow joint. So this is found in the knee or elbow. So here we have between humerus and ulna as one of the examples. So that means hinge joint here is for elbow joint. Okay. That means between humerus and ulna. So the correct match for 2 will be number 4. Third one is pivot joint. Now, where is pivot joint found? Pivot joint is found between the atlas and axis. So the correct match for 3 will be number 2. And the last one is saddle joint. And saddle joint is found between carpals and metacarpals of thumb. So the correct match for 4 will be the first one. So which option should be the correct one? It is only the first option where the match for A is 3, B is 4, then for the third one is second and 1. Okay, so this is the correct answer. Option A will be the correct answer. Question number 29 is from the chapter Locomotion and Movement, and the question is, which of the following diseases is an autoimmune disorder? Okay, options are myasthenia gravis, arthritis, osteoporosis, and gout, and the correct answer is option A, that is myasthenia gravis. Okay, so this is an autoimmune disorder that affects the neuromuscular junction. Okay, so it affects the neuromuscular junction, and it leads to fatigue, weakening, and paralysis of the skeletal muscles. Okay, it leads to fatigue, weakening, and paralysis of skeletal muscles. Now, what is arthritis? Arthritis is an inflammation of joint. Okay, so let's write it down here. Arthritis is inflammation of the joints. Inflammation of joints. Next option is osteoporosis. Osteoporosis. So this is actually an age-related disorder, okay? And in this what happens? The bone mass is reduced and the chances of fractures is increased, okay? So let's write it down here. It is an age-related disorder. It is characterized by decreased bone mass and increase chances of fractures. And then what is the cause of osteoporosis? Cause is the decreased levels of estrogen. Okay, so the cause is decreased level of estrogen. This is a very common cause of osteoporosis. Now what else is there in options? Gout. Okay, so if we talk about gout, it is also the inflammation of joints, but the reason is the accumulation of uric acid crystals. Okay, so this is the inflammation of joints due to accumulation of uric acid crystals. Okay, so this was all about this question. Question number 30 is from the chapter Chemical Coordination and Integration, and the question is, artificial light, extended work time and reduced sleep time disrupt the activity of, options are thymus gland, pineal gland, adrenal gland and posterior pituitary gland, and the correct answer is option B, that is pineal gland. So where is pineal gland located? It is located on the dorsal side of forebrain. Okay, so pineal gland is located on the dorsal side of forebrain. And what is the function of pineal gland? It secretes a hormone known as melatonin. So secretes melatonin hormone. So this hormone is very important because it performs a variety of functions. The most important function is the regulation of 24-hour rhythm of our body. Okay, so 24-hour rhythm, that is also known as diurnal rhythm, right? Diurnal rhythm or circadian rhythm of our body. For example, it maintains the sleep-wake cycle and temperature of the body. Okay, now so this is the most important function, but other than this it also performs many other functions. For example, it influences metabolism, then pigmentation. This is also an important function of melatonin hormone. Then menstrual cycle, as well as defense mechanism of our body. Okay, so this was all about the pineal gland. Question number 31 is again from the chapter Chemical Coordination and Integration, and the question is, which of the following conditions will stimulate parathyroid gland to release parathyroid hormone? Option A, fall in active vitamin D levels. Option B, fall in blood calcium levels. Option C, fall in bone calcium levels. And the last option is rise in blood calcium levels. And the correct answer is option B, fall in blood calcium levels. Okay, so let's see where parathyroid gland is located. Suppose this is our vocal cord. This is vocal cord and this is the trachea or windpipe. Okay, this is trachea. So we know that somewhere here we have our thyroid gland, isn't it? So this shaded portion is the thyroid gland. But we have to talk about the parathyroid gland. So this is located just on the opposite side of the or the back side of the thyroid gland. Okay, so let's draw the back side as well. This is our trachea portion. This is vocal cord. And here we have thyroid gland on the front side. Okay. And this is the back side. So we can see there are two lobes of thyroid gland. So one pair of parathyroid gland is located in each lobe. Okay. So two parathyroid gland in each lobe. So this is parathyroid gland. These four in fact. Now, what is the function of parathyroid gland? It releases a hormone that is known as parathyroid hormone. Okay. So the hormone released by hormone released by parathyroid gland is parathyroid hormone, and in short it is written as PTH. Okay. Now this parathyroid hormone is a hypercalcemic hormone. Hypercalcemic hormone. That means it increases the calcium level in our blood. Increases calcium level in the blood. Okay. How does it do so? It stimulates the reabsorption of calcium ion by renal tubules, and also it increases the calcium absorption from digested food. Okay. So this way it helps in increasing the calcium level in the blood along with thyrocalcitonin hormone. Okay. So according to this question, we have to see that which condition will stimulate the release of parathyroid hormone. Okay, so obviously it helps in increasing the calcium level. That means it will be required when there is lack of calcium in the blood, or we can say when there will be low level of calcium in the blood. Okay, so fall in blood calcium level will stimulate the parathyroid gland to release parathyroid hormone so that it can increase the calcium level in the blood. Okay, therefore option B is the correct answer. Question number 32 is from the chapter Reproductive Health, and here we have to find the correct statement out of the given four statements. So the first statement says IUDs once inserted need not be replaced. So what are IUDs? IUDs stand for intrauterine devices, right? Intrauterine devices. And these are one of the very common contraceptive devices that are used specifically in females, as these are inserted inside the uterus. Okay, so the statement says that they need not be replaced once they are inserted. That is not true, because they have their expiry period after which they need to be removed, and in case they are required even after the expiry period, then they need to be replaced in that case. Okay, so this is the incorrect statement, but we have to find out the correct statement. So let's move to the second statement. It says IUDs are generally inserted by user herself. That is again incorrect, because in order to insert the IUDs, doctors or expert nurses are required. Okay, so doctors or expert nurses insert the IUDs. Okay. Now let's move to the third statement. It says IUDs increase phagocytosis of sperms in the uterus. This is a correct statement, because IUDs are spermicidal. Spermicidal. Not all IUDs. There are different types of IUDs. So most of them are spermicidal. Okay. Let's see what are the types of IUDs. Presently, three different types of IUDs are being used. One of them is non-medicated IUDs. Non-medicated IUDs. Second one is copper releasing IUDs. And the third one is hormone releasing IUDs. Okay. Examples, one of the example of non-medicated IUDs is Lipis loop. Okay. Lipis loop. The example of copper releasing IUDs can be copper T, copper 7 and multi-load 375. And what are the example of hormone releasing IUDs? These are LNG20, LNG20 and progesterone. Progesterone. So we have seen that IUDs increase the phagocytosis of sperms in the uterus. And if we talk about copper releasing IUDs, then they also perform special function of suppressing the sperm's motility. Okay, so they suppress sperm motility as well as their fertilizing capacity. Okay, fertilizing capacity. Then what do hormone releasing IUDs do? They make the uterus unsuitable for implantation. Okay, so they make uterus unsuitable for implantation. Also, they make the cervix hostile to the sperms. Okay, to sperms. So it was all about the IUDs. Okay. So here we can see that the correct statement is the third one. That is option C is the correct one. The last statement is also incorrect. That is IUDs do not suppress gametogenesis, because gametogenesis means what? Formation of gametes. Formation of gametes. But we haven't seen that they prevent the formation of gametes. Gametes are the sperms or ovum. So these are already formed. The IUDs just suppress their movement or their fertilizing capacity, or they make the environment unsuitable for fertilization. Okay, they do not prevent the gametogenesis. So the only correct statement will be the third statement. Now question number 33 is again from the chapter Reproductive Health, and the question is, which of the following sexually transmitted diseases do not specifically affect the reproductive organs? Okay, so first of all, what are sexually transmitted diseases? As the name suggests, these are the diseases or infections that are transmitted through sexual intercourse. Okay, and these are collectively known as sexually transmitted diseases, or in short, these are written as STDs. Other names are also given to such diseases, for example, venereal disease, or in short VD, and reproductive tract infections, that is RTI. Okay, now let's see what are the examples of such diseases. There are many examples. First one is gonorrhea. Gonorrhea. Then we have syphilis. Then genital herpes. Genital herpes. Genital warts. Then chlamydiasis. Chlamydiasis. Trichomoniasis. Trichomoniasis. And then there are two very well known diseases. First one is hepatitis B and the second one is HIV AIDS. Okay, so you must have heard about these two diseases at least. So here we must remember one thing, that hepatitis B and HIV AIDS are the diseases which do not specifically affect the reproductive organs. Okay, hepatitis B affects which part? Liver. And HIV AIDS, as we all know, that it affects the immune system of our body. Therefore, in option C, these two are mentioned. That is why this option will be the correct answer. So, these two diseases do not specifically affect the reproductive organs of the body. Okay. Another important thing that we should remember, that genital herpes, then hepatitis B and HIV/AIDS. These three are non-curable, and all the others are curable diseases. Okay. So, I hope you understood this. Now question number 34 is from the chapter Animal Kingdom, and here we have to match the names of genera with their respective phylum. Okay, so the first one is Ophiura. Ophiura. What is the common name of Ophiura? It is commonly known as brittle star. Brittle star, and the phylum that it belongs to is Echinodermata. Echinodermata. And one of the most important characteristics of echinoderms is the presence of water vascular system. Water vascular system that is responsible for many things. For example, locomotion, food intake and transport, as well as respiration. Okay. So the correct match for A will be number 3. Echinodermata. Now let's come to the second one. It's Physalia. Physalia again has a common name, and its common name is Portuguese man of war. Portuguese man of war. And which phylum does it belong to? It belongs to the phylum Coelenterata. And as you might be aware, the coelentrates are also known as cnidarians. Okay, so Coelenterata is also known as cnidaria. And the name is given due to the presence of a special type of cells known as cnidocytes or cnidoblasts. Cnidocytes or cnidoblasts. Okay, so the correct match for B will be number 4. Now the third one is Pinctada. Pinctada. What is the common name of Pinctada? It is known as pearl oyster. Pearl oyster, and it belongs to the second largest phylum of animal kingdom, that is Mollusca. Mollusca. Okay. So the match for C will be number 1. And the last one is Planaria. So Planaria have a dorsoventrally flattened body. Therefore, these are also known as flatworms. Okay. Flatworms, and the phylum is known as Platyhelminths. Platyhelminths. And one of the most important characteristics of platyhelminths is that they have very high regeneration capacity. High regeneration capacity. So, out of the given options, which one will be the correct one? Option B. Okay, this shows the correct matches. Question number 35 is again from the chapter Animal Kingdom, and the question is, which of the following animals are true coelomates with bilateral symmetry? Options are adult echinoderms, Aschelminthes, Platyhelminths and Annelids. So what does the term coelomates mean? Coelomates are the animals that possess coelom. Okay, and what is coelom? Coelom is the body cavity that is lined by mesoderm. So let's write it down here. Body cavity that is lined by the mesoderm is known as coelom. Now, depending on whether an animal possesses coelom or not, they are classified into two groups. First one is coelomates and the other is acoelomates. So as the name suggests, coelomates are those animals which possess coelom, and acoelomates are those which do not have coelom. Okay, and the examples of acoelomate animals is platyhelminths. Platyhelminths. So this option will naturally be eliminated because here it is asked about the true coelomates, but platyhelminths is acoelomate. Okay, so this option is eliminated here. Now again, coelomates are classified into two groups. First group is true coelomates and the other is pseudocoelomate. Okay, pseudocoelomates. So in true coelomates, the mesoderm lines the body cavity in a regular manner. But in pseudocoelomates, mesoderm is not present in regular manner. Instead, it is scattered like pouches. Okay, so mesoderm is present as scattered pouches here between ectoderm and endoderm. Ectoderm and endoderm. Example of pseudocoelomates is Aschelminthes. So Aschelminthes is not a true coelomate. Okay, so again Aschelminthes, that is option B, is also eliminated. So we have two options left, option A and option D. That means adult echinoderms and annelids. So both of them are true coelomates. But here it is also mentioned that we have to find out the true coelomate with bilateral symmetry. Okay, so here the correct option will be option D, that is annelids, because adult echinoderms do not have bilateral symmetry. These have radial symmetry. Okay, which kind of symmetry? Radial symmetry. So option D will be the correct answer. Question number 36 is from the chapter named The Living World, and the question is the contrasting characteristics generally in a pair used for identification of animals in taxonomic key are referred to as, options are lead, couplet, doublet and alternate, and the correct option is option B, that is couplet. So this is a direct question. What is a taxonomic key? As mentioned in the question, it is an aid that is used for identification of plants and animals. Okay, so taxonomic key is an aid that is used for identification of plants and animals, and it is based on contrasting characters. Based on contrasting characters, that means opposite characters, and then these have to be analyzed and one choice has to be made between the two opposite characters. Okay, so it represents the choice made between two opposite characters. So we can say that this is analytical in nature because it has to be analyzed first, right? So analytical in nature. And the contrasting characters are known as couplets. So option B will be the correct answer. Here you can also remember that each statement that is made in the key is known as lead. Okay, so the first option, that is lead, is actually each statement made in the key. Okay. Now let's move to question number 37. It is from the chapter Structural Organization in Animals. And here we have to match the given cell structures with their characteristic features. Okay. So the first one is tight junctions. It is one of the three main types of cell junctions, right? And what is the function of tight junctions? It prevents the leakage across the cells. Okay. So the correct match for A will be number 3. It establishes a barrier to prevent leakage of fluid across epithelial cells. So the correct match for A will be number 3. Second one is adhering junction. This is second type of cell junctions and it helps in cementing different cells together. Okay, so the match for B will be number 1. Cement neighboring cells together to form sheet-like structure. So the match for B is number 1. Third one is gap junction, and what does gap junctions do? They help in communication. So the correct match for C will be number 4. Cytoplasmic channels to facilitate communication between adjacent cells. Okay, so communication is the main word here. So the correct match for C is number 4. Last one is synaptic junction and the only option left is second one. So the synaptic junction transmit junction information through chemical to another cells. So the correct match for D will be number 2. So which is the correct option out of the given options? Option C. It has correct matches here. Question number 38 is again from the chapter Structural Organization in Animals. And the question is, which of the following statements is incorrect? So the first statement says cockroaches exhibit mosaic vision with less sensitivity and more resolution. So this itself is the incorrect statement. The first part of the statement is correct, that cockroaches exhibit mosaic vision. So this is true. But mosaic vision has more sensitivity and less resolution, just opposite of what is mentioned here. Okay, so it has more sensitivity and less resolution. And this type of vision is common during night. Therefore, this is also known as nocturnal vision. Okay, nocturnal vision. So we have already got the incorrect statement, but still let's go through other statements as well. The second statement says a mushroom shaped gland is present in the 6th and 7th abdominal segments of male cockroach. So this is a correct statement. Okay, so this mushroom shaped gland that actually acts as an accessory reproductive gland. Okay, so this acts as an accessory reproductive gland. Now let's come to the third statement. It says a pair of spermatheca is present in 6th segment of female cockroach. So this is also a correct statement. Okay, and it opens into the genital chamber. Now the last statement says female cockroach possesses 16 ovarioles in the ovaries. So this is also a correct statement. 16 ovarioles, that means 8 pairs of ovarioles. Okay, 8 pairs of ovarioles, and that is also known as ovarian tubules. Ovarian tubules which containing a chain of developing ova. So the correct answer for this question is option number A. Okay. Question number 39 is from the chapter Breathing and Exchange of Gases, and the question is select the correct statement. So here we have to select the correct statement, right? The first statement says expiration occurs due to external intercostal muscles. So what is expiration? It is one of the processes in breathing, and in this the air is expelled out of the lungs into the atmosphere, right? So let's see what happens during breathing. During the process of breathing, two main steps are there. First one is inspiration and the second one is expiration. So in case of inspiration, what happens? Air from the surrounding enters into the lungs. And why does this happen? This happens due to the contraction of diaphragm and external intercostal muscles. Okay. So during inspiration, what happens? Contraction of diaphragm and external intercostal muscles takes place. Okay, So what happens when the diaphragm and external intercostal muscles contract? When they contract, space is created. Okay, more space is created, and due to that, the volume, that is thoracic volume, increases. Okay, thoracic volume increases, and when the volume increases, the pressure will automatically decrease. Okay, so the pulmonary pressure decreases, and this pulmonary pressure becomes less than the atmospheric pressure. Okay, so we know that the air will move from higher pressure to the lower pressure. Therefore, the air will move from the atmosphere into the lungs. Okay, this happens in case of inspiration, and the opposite will happen in case of expiration. So in this case, instead of contraction, the diaphragm and intercostal muscles will relax and they will come back to their original position. Okay, so in this, what happens? Relaxation of diaphragm, relaxation of diaphragm and intercostal muscle, that is external intercostal muscle, takes place, right? So they come back to their original position, and this time they will occupy the position they had previously occupied. So the thoracic volume will decrease this time. Okay, and when the volume will decrease, what will happen to pressure? The pulmonary pressure will increase. Pulmonary pressure will increase and it will become more than the atmospheric pressure, right? So what will happen? This time the pressure is more inside the lungs, so it will move out towards the atmosphere. Okay, so here air moves out. Now, let's come back to the question and see what are the given options. The first option is that expiration occurs due to external intercostal muscles. So, yes, external intercostal muscle is there, but diaphragm is also involved. Okay, but in this statement, diaphragm is not mentioned. So, this will not be the correct statement. Second statement says that intrapulmonary pressure, that means the pressure inside the lungs, is lower than the atmospheric pressure during inspiration. So come to here, inspiration, the pulmonary pressure is lower than the atmosphere. So this is true. So yes, this is a correct statement. Okay. Third statement says inspiration occurs when atmospheric pressure is less than the intrapulmonary pressure. So atmospheric pressure is not less, okay, it is more than the pulmonary pressure. So this is not true. And the last statement says expiration is initiated due to contraction of diaphragm. No, this is initiated due to relaxation, right? Inspiration is initiated due to contraction and expiration is initiated due to the relaxation. So the only correct statement is statement B. So option B will be the correct answer. Now again, this question is from the same chapter, breathing and exchange of gases. Okay, and the question is the maximum volume of air a person can breathe in after a forced expiration is known as. Options are expiratory capacity, vital capacity, inspiratory capacity and total lung capacity, and the correct answer is option B, that is vital capacity. Okay, so this is a direct definition based question. Vital capacity is the maximum volume of air a person can breathe in after a forced expiration. What is expiratory capacity? This is the total volume of air that can be expired. Okay, so the total volume of air that a person can expire after a normal inspiration. Okay, after a normal inspiration. And what is inspiratory capacity? This is the total volume of air that a person can inspire after a normal expiration. And the last one is total lung capacity. So this is the total volume of air that can be accommodated in the lungs. So the total volume of air that can be accommodated in the lungs at the end of a forced inspiration. So the maximum volume of air that can be accommodated is the total lung capacity. Okay. Okay, now let's move to question number 41. It is from the chapter Body Fluids and Circulation. And the question is, all the components of the nodal tissue are auto-excitable. Why does the SA node act as the normal pacemaker? First option is SA node has the lowest rate of depolarization. Second option is SA node is the only component to generate the threshold potential. Third option says only SA node can convey the action potential to the other components. And the last option is SA node has the highest rate of depolarization. Okay. So what is SA node? SA node stands for sinoarterial node. Okay. Sinoarterial node. Now, as mentioned in the question that all the components of the nodal tissue are auto-excitable. Auto-excitable. That means they can function on their own, or we can say they can generate the action potential on their own. Generate the action potential without any external stimuli. Okay, without any external stimuli. Now, if we talk about the sinoarterial node, then we should remember that this is the component that generates the maximum number of action potentials. Okay, so maximum number of action potentials, about 70 to 75 per minute, are generated by the sinoarterial node. Okay, so due to this high number of action potentials, this sinoarterial node is able to initiate as well as maintain the rhythmic contraction of the heart. Okay, so we can say that it sets the pace of activities of the heart. And this is the reason why it is known as the pacemaker. Okay. So maximum number of action potentials are generated. That means it will have the highest rate of depolarization, right? So option D will be the correct answer. SA node has the highest rate of depolarization. Question number 42 is again from the chapter Body Fluids and Circulation, and the question is, a specialized nodal tissue embedded in the lower corner of the right atrium close to arterioventricular septum delays the spreading of impulses to heart apex for about 0.1 seconds. The delay allows, options are, blood to enter aorta, the ventricles to empty completely, blood to enter pulmonary arteries, and the atria to empty completely. Okay. So let's first understand the question and then we'll come back to the options. We know that the human heart has four chambers, two atria and two ventricles. Okay. So suppose this is right aorta, this is left aorta. Similarly, this is right ventricle and this is left ventricle. Right. Now, as mentioned in the question, a specialized nodal tissue is embedded in the lower corner of right atrium. So this is the right atrium. Okay. So lower here, lower corner. Okay. And this is the arterioventricular node. So we are talking about the arterioventricular node here. At this point, you will have the sinoarterial node that we have discussed about in the previous question. Now, the role of AVN or the ventricular node comes after the blood enters into this aorta. Okay, so the first option will not be the correct one because the delay will not allow the blood to enter aorta, because at this point the blood has already entered the aorta. Okay, after that only the role of AVN comes. Second one is the ventricles to empty completely. So till now the blood has not moved into the ventricle, so ventricle has not come into scene now. So this is also incorrect option. Third one is blood to enter pulmonary arteries. Again, the pulmonary arteries have no role to play at this point. Then the last one is arteria to empty completely. Yes, this is the correct option because at this point the blood is there in this aorta, okay, or you can say arteria, and at this point the blood will be flowing out of the artery into the ventricle, right? Now when it will delay the spreading of impulses to heart apex, in that case more blood will be able to escape from this arteria, therefore the arteria will be emptied completely. Okay, so option D will be the correct answer. Question number 43 is from the chapter Excretory Products and Their Elimination. And here we have to match the parts of a nephron with their function. So what is a nephron? Nephron is the functional unit of a kidney. Okay, so let's write it down here. Functional unit of kidney is known as nephron. Now this nephron has two main parts, glomerulus and renal tubule. Now we'll discuss renal tubule in detail because different parts of renal tubules are mentioned in this question. Okay, so the renal tubule starts with the Bowman's capsule, which looks something like this. This is Bowman's capsule, and inside this Bowman's capsule glomerulus is present. Now as you can see here that the renal tubule has different parts or different shapes. First it goes straight, then it goes downward, then it goes upward. So this U-shaped part is known as Henle's loop or loop of Henle. Okay, this loop of Henle has two limbs. First one is descending limb that is going downward, and the second part is ascending limb that is moving upward. Okay, this is ascending limb. And this last part, straight part, is DCT, that is distal convoluted tubule. The first straight part, this one is PCT, that is proximal convoluted tubule. Okay, so let's see what are the functions of each of them. First we'll discuss about PCT, proximal convoluted tubule. Okay, P for proximal. So here maximum reabsorption takes place. Maximum reabsorption. Okay, about 70 to 80% of the electrolytes are reabsorbed in this part. Other than that, water and organic nutrients are also reabsorbed. So the correct match for proximal will be what? Number 4, reabsorption of tubule ion, water and organic nutrients. So here in option number B and C, we can see that B is matched with 4. So we'll see number 1 now, descending limb. Okay, so this is the next part, descending limb. What happens in descending limb of loop of Henle? This limb is permeable to water. Permeable to water. Therefore, water is reabsorbed here. Okay. So, convoluted water is reabsorbed. Convoluted water is reabsorbed in this part. This Now this descending limb is impermeable to electrolytes, so only water is reabsorbed. So the correct match for A, that is descending limb, will be number 2, reabsorption of convoluted tubule water only. So A will be matched with 2. So in option B, we can see that A is matched with 2. So option B will be the correct answer. Still we'll discuss other parts also. Third one is ascending limb. Okay, ascending limb is just the opposite of descending limb. That means this is permeable to electrolytes and it is impermeable to water. Okay, permeable to electrolytes. Therefore, salts are reabsorbed here. Okay, so reabsorption of salts will take place here. Okay, reabsorption of salts or electrolytes will take place. So the correct match for C will be number 1. Okay, so here you can see C is matched with number 1. And the last one is distal convoluted tubule, this DCT. So here conditional reabsorption takes place. So the correct match for D will be number 3, conditional reabsorption of sodium ion and water. Okay, so option B is the correct answer. Question number 44 is again from the chapter excretory products and their elimination. And the question is match the items in column 1 with those in column 2. So the first thing that is mentioned in column 1 is podocytes. Podocytes. So what are podocytes? See, in the previous question we discussed that the renal tubule starts with the Bowman's capsule, isn't it? And inside the Bowman's capsule lies the glomerulus. Anyways, why are we discussing about Bowman's capsule? Because podocytes are nothing but the epithelial cells of Bowman's capsule. Okay, so let's write it down here. These are epithelial cells of Bowman's capsule. Now these are arranged in an intricate manner, leaving some minute pores, or you can say leaving some small spaces which are known as filtration slits or slit pores. Okay, so the minute spaces here, minute spaces are there which are known as filtration slits or slit pores. Okay, so here blood is filtered so finely that almost all the constituents of the plasma membrane can easily pass through these pores, okay, except protein, because protein is much larger as compared to other constituents. Therefore, the process is known as ultrafiltration. Okay, so the ultrafiltration takes place here. Therefore, the correct match for A will be number 4, that is filtration slits. Okay, so here either option C or D can be the correct answer because here A is matched with number 4. Now let's come to the second one, that is protonephridia. Protonephridia. So protonephridia is nothing but the excretory structures. These are excretory structures in some organisms. Okay, structures in platyhelminths, then rotifers. Some annelids also have protonephridia as the excretory structures. Some annelids, but not all the annelids, okay? Only some annelids. And cephalochordates also have protonephridia. Cephalochordates. And in cephalochordate, that is Amphioxus. Okay, Amphioxus is a cephalochordate. So here annelids and Amphioxus both are mentioned. So protonephridia will be matched with Amphioxus because not all annelids have protonephridia, only some annelids have. Okay, so it will be matched with number 3, that is Amphioxus. Now we come to the third one, that is nephridia. So most of the annelids have nephridia. Okay, so nephridia will be matched with annelids. Okay, and then earthworms also have nephridia. So anyways, here it has to be matched with annelids. So it is quite clear that B will be matched with number 3 and C will be matched with number 2. So option C will be the correct answer. Last one is renal calculi. Renal calculi. So we'll see this one also. Here, renal calculi are nothing but the stones or insoluble mass of crystallized salts. Crystallized salts such as oxalates. So renal calculi will be matched with crystallized oxalates, that is number 1. So it is clear that C option is the correct answer. Question number 45 is from the chapter Neural Control and Coordination. And the question is, which of the following receptors are specifically responsible for maintenance of balance of body and posture? Options are basilar membrane and otoliths, then hair cells and organ of Coti, then tectorial membrane and macula, and the last one is crista ampullaris and macula. And the correct answer will be option D, that is crista ampullaris and macula. Okay, so let's see, we know that the ear has three parts: outer ear, then middle ear, and then the innermost, that is inner ear, right? Now this inner ear has a complex system known as vestibular apparatus. Okay, so it contains vestibular apparatus, and this vestibular apparatus has some specific receptors known as crista and macula. Okay, these are specific receptors of vestibular apparatus which are responsible for maintenance of balance of body and posture. Okay, balance of body and posture. So option D is the correct answer. Question number 46 is from the chapter The Living World, and the question is, which of the following is against the rules of ICBN? What is ICBN? It means International Code for Botanical Nomenclature. So there are some rules that we have to follow while naming an organism or a species. Okay. So here botanical nomenclature, that means this is related to plants. So let's see, the first one says that handwritten scientific names should be underlined. This is a correct rule. Okay, this rule is followed, that whenever we write the scientific names, then we underline them separately. So first we write the generic name, and then we write specific name. Okay, and then we underline them separately, not a single line, two separate lines. Okay, then the second option is that every species should have a generic name and a specific epithet. So this is also correct. Okay, then the third one is scientific names are in Latin and should be italicized. Of course, this is also correct. Okay, no matter what the origin of the name is, it has to be Latinized. Okay, and it has to be written in italics. So this is also correct. The last one says generic and specific names should be written starting with small letters. So according to this, both should be starting with small letters, which is not true, because a generic name starts with capital letters, only specific name starts with small. Okay, so generic name starts with capital letter and specific name start with small letter. So this option is against the rules of ICBN. So option D will be the correct choice here. Question number 47 is from the chapter Biological Classification, and the question is, mad cow disease in cattle is caused by an organism which has, options are inert crystalline structure, then abnormally folded protein, then free RNA without protein coat, or free DNA without protein coat. So the correct answer will be option B, that is abnormally folded proteins. So the organisms which have abnormally folded proteins are known as prions. Okay. So here we are talking about the prions, and these prions are responsible for the transmission of certain infectious neurological diseases. Okay. Transmission of certain infectious neurological diseases. And one of the most notable examples of such a disease is bovine spongiform encephalopathy. Okay, bovine spongiform encephalopathy. In short, it is written as BSE. Okay, and it is commonly known as mad cow diseases. Mad cow disease, and it is common in cattle. Okay. So therefore, option B will be the correct answer, that mad cow disease is caused by an organism which has abnormally folded proteins, and these organisms are known as prions. Okay. Question number 48 is again from biological classification, and the question is, which of the following statements is correct? So we have to find out the correct statement. First statement says lichens do not grow in polluted areas. So this is a true statement. Okay. So first option itself is the correct statement, but still let's go through other statements as well. Second statement says algal component of lichens is called mycobiont. So we know that the lichens remain in symbiotic association between algal component and fungal component. Okay, so algae and fungi. Algal component are known as phycobiont, and fungal component are known as mycobiont. So here the second statement says that algal component of lichens is called mycobiont. That is not true because these are known as phycobiont, right? And the third one says that fungal component of lichens is called phycobiont. This is again incorrect because the fungal component are known as mycobiont. So just the opposite is mentioned here. The last statement says lichens are not good pollution indicators, which is again incorrect because they are good pollution indicators as they do not grow in polluted areas. Okay, so this way they act as pollution indicators. So if you put a lichen in some area that is polluted, they will not grow. So this way they will indicate that there is pollution in this area. So option A will be the correct answer. Question number 49 is also from the chapter Biological Classification, and the question is match the organisms in column 1 with their habitats in column 2. So the first organism that is mentioned in column 1 is halophiles. So halophiles are type of archaebacteria, right? And these are found in extremely salty areas. So the correct match for A will be number 4, that is salty area. Then the second one is thermoacidophiles. So we know that the term therm is related with heat. Okay. So thermoacidophiles are found in hot areas such as hot springs. So the correct match for B will be number 1. That is hot springs. Okay. The third one is methanogens. So methanogens are those bacteria which produce methane gas, okay, which they actually help in the production of methane gas from the dung of ruminant animals, okay, and these are found in the guts of ruminant animals, okay. So this is already matched here. C is matched with number 3. And the last one is cyanobacteria. So cyanobacteria are type of eubacteria. Okay. Others were examples of archaebacteria, and this is example of eubacteria, and this is found in aquatic environments. So the correct match for D will be number 2. So out of the given options, option A will be the correct answer. Question number 50 is from the chapter Anatomy of Flowering Plants, and the question is, in the dicot root, the vascular cambium originates from option A, tissue located below the phloem bundles and a portion of pericycle tissue above protoxylem. Option B, cortical region. Option C, parenchyma between endodermis and pericycle. And the last option is intrafascicular and interfascicular tissue in a ring. And the correct answer is option A, that is tissue located below the phloem bundles and a portion of pericycle tissue above protoxylem. Okay, so here the word pericycle is important. So we know that the outermost layer is known as epidermis, right? So suppose this is epidermis. We know that there are root hairs on epidermis. So these are root hairs. Then if we move inside the epidermis, the next layer of cells is known as cortex. So these are cortical cells, okay? Cortex. And suppose till here we have cortex. So the innermost layer of the cortex is known as endodermis, okay? So this is the endodermis, suppose. Endodermis. Now, if we move inside the endodermis here, we have a layer of parenchymatous cells known as pericycle. Okay. So these are pericycle and these are parenchymatous cells. Okay. Parenchymatous cells. And these are important as the lateral roots as well as vascular cambium originate from this portion. Okay. So option A will be the correct answer. Question number 51 is from the chapter Morphology of Flowering Plants, and the question is, which of the following shows world phyllotaxy? Options are mustard, China rose, Alstonia and Calotropis. So what is meant by the term phyllotaxy? Phyllotaxy is the pattern of arrangement of leaves on the stem. Okay, pattern of arrangement of leaves on the stem or branch. Okay. Now, there are mainly three types of phyllotaxy. First one is alternate type, alternate phyllotaxy. In this, what happens? Only single leaf arises at each node. Okay. So, a single leaf at each node. Examples of alternate type of phyllotaxy will be your China rose, then mustard and sunflower. Okay. So these have alternate phyllotaxy. So obviously China rose is not the correct answer and mustard is also not the correct answer because it is asked about world phyllotaxy, right? So we'll see further. Second type of phyllotaxy is known as opposite. Opposite phyllotaxy is when two leaves, or you can say a pair of leaves, okay? A pair of leaves are present at each node. In this case, both the leaves will be opposite to each other, right? Example for this will be Calotropis. Calotropis. So this is also not the correct answer here because this is the example of opposite type of phyllotaxy. And the third and the last one is world phyllotaxy, which is asked in this question. So it happens when more than two leaves are present at each node. Okay, more than two leaves. An example is Alstonia. So the correct answer will be option C, Alstonia. Now question number 52 is from the chapter Anatomy of Flowering Plants, and the question is, regeneration of damaged growing grass following grazing is largely due to. Options are lateral meristem, apical meristem, intercalary meristem and secondary meristem. And the correct answer will be option C, that is intercalary meristem. Okay, so intercalary meristem occur between the matured tissues. Okay, so they occur in between mature tissues, and these are found in grasses, and when the grasses are grazed by the animals, then these also help in regeneration of the damaged parts. Okay, so option C is the correct answer. Question number 53 is from the chapter Morphology of Flowering Plants, and the question is, bicarpellary ovary with obliquely placed septum is seen in, options are Brassica, Aloe, Solanum and Sesbania, and the correct answer is option C, that is Solanum. Okay, so let's discuss the options one by one. Brassica belongs to the family Brassicaceae, right? And it is clear from the name also. And we know that Brassicaceae family have bicarpellary ovary. Bicarpellary ovary. Okay. Next one is aloe. Aloe belongs to which family? Liliaceae family. And Liliaceae family have tricarpellary ovary, not bicarpellary. Okay. So this clearly is not the correct answer, because option is asked about bicarpellary ovary. Okay. So this is not true. Next one is Solanum. Solanum belongs to Solanaceae family. And Solanaceae family also have bicarpellary ovary. The last one is Sesbania. Sesbania belongs to which family? It belongs to Fabaceae family. And how many carpels are there in case of Fabaceae? Only one. So the ovary is monocarpellary here. These have monocarpellary ovary. So this is also not the correct answer, because we have to find out the bicarpellary ovary only. So either option A or option C can be the correct answer. But the question also mentioned that obliquely placed septum is there. So in case of Brassica, or you can say in case of Brassicaceae family, there is a false septum. Okay, the septum is not true. So false septum is there, therefore this is also not correct answer. In case of Solanum, there is a true septum, and what kind of septum is there? Obliquely placed septum. Okay, obliquely placed septum. Therefore option C is the correct answer. Now question number 54 is from the chapter Sexual Reproduction in Flowering Plants, and the question is, which is the most common type of embryo sac in angiosperms? Options are tetrasporic with one mitotic stage of divisions, monosporic with three sequential mitotic divisions, monosporic with two sequential mitotic divisions, and the last one is bisporic with two sequential mitotic divisions. So the correct answer will be option B, that is monosporic with three sequential mitotic divisions. Now let's see what it means. First of all, what is embryo sac? Embryo sac is the female gametophyte of the plant. Okay, so it is female gametophyte. Where is it located? It is located in the nucellus of the ovule. Okay, located in the nucellus of the ovule. Now, here we should be aware with one more term, that is megasporogenesis. Okay, megasporogenesis. That means genesis of megaspores, or you can say this is the process of formation of megaspores. Okay, so this is the formation of megaspores from the megaspore mother cell, that is MMC in short. Okay, MMC means megaspore mother cells. So what happens? It starts with the meiotic division of the megaspore mother cell. So here we have, suppose, megaspore mother cell. This megaspore mother cell undergoes what kind of division? Meiotic division. Okay, meiotic division, that means it undergoes meiosis. And how many megaspores will be formed after meiotic divisions? 4 megaspores will be formed. So 4 megaspores are there. Now, out of these 4 megaspores, 3 megaspores degenerate. Okay, 3 degenerate. And only 1 megaspore remains functional. And the embryo sac develops from this one functional megaspore. Okay, so it will be monosporic because out of 4 only one spores leads to the formation of embryo sac. Okay, bisporic is not correct because 2 spores are not developing into embryo sac. Okay, so tetrasporic is also not true, only monosporic is possible. Now we'll see how many sequential mitotic divisions take place. So this is the functional megaspore. Okay, this is functional megaspore. So total 3 mitotic divisions takes place. Okay, so this is the first mitotic division. It leads to the formation of 2 megaspores. Then again one more. Then these will be doubled. Then one more. Okay, so this is second mitotic division, and then this is third mitotic division, and here 4 megaspores this side and 4 megaspores this side. Okay, so 3 sequential mitotic divisions take place. Therefore option B will be correct, that it is monosporic with 3 sequential mitotic divisions. Question number 55 is from the chapter Molecular Basis of Inheritance, and the question is, from the following, identify the correct combination of salient features of genetic code. Option A, universal, non-ambiguous and overlapping. So this is not true because this is not overlapping. Okay. Option B, degenerate, overlapping and commaless. Again, it is not overlapping, so this is not true. Then universal, ambiguous and degenerate. So this is not ambiguous, so this is also not true. The last one is degenerate, non-overlapping and non-ambiguous. Yes, this is true. Okay. So what is meant by the term degenerate? Degenerate. It means that some amino acids are coded by more than one codon. Okay. Some amino acids are coded by more than one codon, or you can say that different codons can code for same amino acid. Okay, so more than one codons can code for one amino acid. Then it is non-overlapping. Non-overlapping, it means that adjacent codons are non-overlapping, or you can say the adjacent codons do not overlap. Then last we have non-ambiguous. Non-ambiguous, that means specific. Okay, so these are specific, that means one codon will code for only one amino acid. One codon codes for only one amino acid. Okay, so option D will be the correct answer. Question number 56 is again from Molecular Basis of Inheritance, and the question is, which scientist experimentally proved that DNA is the sole genetic material in bacteriophage? Options are Beadle and Totem, Michelson and Stahl, Hershey and Chase, and Jacob and Monod. So the correct answer is option C, that is Hershey and Chase. Okay, So they gave the unequivocal proof of DNA being the genetic material. Okay. So let's write it down here. Unequivocal proof that DNA is the genetic material. In the year 1952. Okay. First one is Beadle and Totem. What did Beadle and Totem do? They proved that genetic code governed the formation of enzymes. Okay. Genetic code govern the formation of enzymes in the year 1941. Second one is Michelson and Stahl. So they performed the separation of DNA by centrifugation. Separation of DNA by centrifugation in the year 1958. And the last one is Jacob and Monod. So they performed what? Elucidation of lac operon. Elucidation of lac operon. And they were the first to elucidate a transcriptionally regulated system. Okay. They were the first to elucidate a transcriptionally regulated system. So this was all about this question. The correct answer is what? Third number according to this question. Okay. Okay, so the same chapter continues here, and the question is, in the process of transcription in eukaryotes, the RNA polymerase 1 transcribes, options are mRNA with additional processing, capping and tailing, then tRNA, 5 srRNA and snRNAs, then rRNAs, 28S, 18S and 5.8S, and the last one is precursor of mRNA, that is hnRNA. So the correct answer is option C. Okay. We know that eukaryotes have at least three types of RNA polymerases in the nucleus. Okay. So eukaryotes have at least three. Okay. It can be more than this also. So at least three RNA polymerases in the nucleus, in addition to the RNA polymerase that are present in the organelles. Okay. Now these three RNA polymerases show clear-cut division of labor. Okay. Division of labor is shown. So RNA polymerase 1 performs the transcription of rRNAs, this one. Third one, 28S, 18S and 5.8S. Okay. RNA polymerase 2 performs transcription of precursor of mRNA, that is hnRNA. So this last one is done by RNA polymerase 2, and RNA polymerase 3 transcribes tRNA. Okay, this second one. Okay, so RNA polymerase 3 transcribe 5 srRNA and snRNA as well as tRNA. Okay, so I hope you understood this. Question number 58 is from the chapter Principles of Inheritance and Variation. And the question is, in which genetic condition each cell in the affected person has three sex chromosomes, that is XXY. Okay. So options are thalassemia, Klinefelter's syndrome, phenylketonuria and Turner's syndrome. And the correct answer is option B, that is Klinefelter's syndrome. Okay. So what happens in this syndrome? It happens due to the presence of an additional copy of X chromosome. Okay. We know that generally a male has what? X and Y. Okay. Generally we have XY. So when an additional X is added, it becomes XXY. Right. So Klinefelter syndrome occurs due to the presence of an additional copy of X chromosome. So generally we have how many chromosomes? 46 chromosomes. Okay, humans have 46 chromosomes, that is 23 pairs of chromosome. Okay, so when one additional X is added, how many chromosomes are there? It becomes 47 chromosomes. Okay, so total 47 chromosomes are there in this syndrome. Okay, with karyotype XXY. Okay, it results in sterility in males. Okay, sterility in males. So this is how option B will be the correct answer. Let's discuss other options as well. First one is thalassemia. Thalassemia occurs due to the lack of hemoglobin. Okay, so the reason is the mutation in the DNA of the cells that make the hemoglobin. Okay, mutation in the DNA of cells that make hemoglobin. Therefore, hemoglobin is not formed. So, lack of hemoglobin causes thalassemia. Okay. Phenylketonuria. So, let's write it down here. Phenylketonuria. This is actually an inborn error of metabolism. Okay. Inborn error of metabolism. So, what happens in our body? Phenylalanine is converted into tyrosine. Okay, so in case of phenylketonuria, the enzyme that converts phenylalanine into tyrosine is lacking. Okay, therefore this conversion is not done. So what happens? Phenylalanine is not converted into tyrosine, therefore it gets accumulated. Okay, so this is accumulated and then it is converted into phenylpyruvic acid and other related substances. Okay, so accumulation in the brain also causes mental retardation. Okay, so this was all about phenylketonuria. Last one is Turner's syndrome. Okay, so let's write it down here. Turner's syndrome. So this syndrome occurs due to the absence of one X chromosome. Okay, in Klinefelter syndrome, an additional X was present. In this case, an X is lacking. Okay, so this is due to the absence of one of the X chromosomes in females. Okay, so we know that in general we have XX chromosomes. Okay, females have XX. So when one X is lacking, it becomes XO. Okay, so this is the karyotype in this syndrome. Okay, so I hope you understood this question as well. Now let's see question number 59. It is from the chapter Molecular Basis of Inheritance. And the question is, what initiation and termination factors are involved in transcription in eukaryotes? Okay, so we know that in case of eukaryotes, transcription involves three steps. Okay, transcription involves three steps. First one is initiation. Then the second one is elongation. And the third one is termination. Now here we have to note that RNA polymerase is not able to catalyze all these three steps. Okay, RNA polymerase is able to catalyze only the process of elongation. Okay, this is done solely by RNA polymerase. But for the process of initiation and termination, RNA polymerase needs to be in association with some factors. Okay, so in case of initiation, RNA polymerase needs to be associated with RNA polymerase associates transiently with initiation factor. Initiation factor, that is sigma. Okay, and in case of termination, RNA polymerase associates transiently with termination factor, and termination factor is rho. Okay, so the initiation factor is sigma and termination factor is rho. Therefore, option A will be the correct answer, sigma and rho respectively. Question number 60 is from the chapter Evolution, and the question is, which of the following statements is correct about the origin and evolution of men? Okay, so we have to find out the correct statement. First statement says agriculture came around 50,000 years back, which is not true because the agriculture came around 10,000 years back. Okay, around 10,000 years back. So this is not the correct one. Second statement says the Dryopithecus and Ramapithecus primates existing 15 million years ago walked like men. So this part is true that they existed 15 million years ago. So this is correct, but they did not walk like men. Okay, this is not true. They walked like gorillas and chimpanzees. Okay, gorillas and chimpanzees, not like men. Okay, third statement says Homo habilis probably ate meat. Not true because they did not eat meat. Okay, then the last statement says Neanderthal men lived in Asia between 1 lakh and 40,000 years back. This is absolutely correct statement. Therefore, option D will be the correct answer. Now, question number 61 is from the chapter Principles of Inheritance and Variation. And the question is the production of gametes by the parents, the formation of zygotes, the F1 and F2 plants can be understood using options are pie diagram, a pyramid diagram, Punnett square and Venn diagram. So the correct answer will be option C, that is Punnett square. Okay, so it looks something like this. Okay, it has boxes. We write the genotypes of parents on two sides. Suppose one of the parents has genotype capital T small t and the other has genotype small t. Okay, so one genotype will be written on one side capital T small t like this, and the second one like this, and then we'll find the genotypes of the progenies. Okay, for example, this small t capital T here, then small t here, small t capital T, and small t. Okay, so this square helps in finding the production of gametes by parents, then the formation of zygotes. Okay, so all these can be understood using this Punnett square. So option C is the correct answer. Question number 62 is from the chapter Cell, the Unit of Life, and here we have to match the organelles that are mentioned in column 1 with their functions mentioned in column 2. Okay, so the first organelle that is mentioned is Golgi apparatus. So what does Golgi apparatus do? It helps in the formation of glycoproteins and glycolipids. Therefore, the correct match for A will be number 3. Okay. Then the second one is lysosomes. Lysosomes help in digesting the biomolecules. Okay. Sometimes it also digests itself. Okay. Therefore, it is also known as suicidal sac. So the correct match for B is number 4. That is, its function is digesting the biomolecules. Then the third one is vacuoles. What do vacuoles do? They trap waste and excretory products. So it will be matched with number 2. And the last one is ribosomes. They help in the synthesis of protein. So the correct match for D will be number 1. Okay. So out of the given options, option A will be the correct answer. Now question number 63 is from the chapter Biomolecules, and the question is prosthetic groups differ from coenzymes in that options are they require metal ions for their activity, then they are tightly bound to apoenzymes, Third option says their association with apoenzyme is transient. And the last one is they can serve as cofactors in a number of enzymes catalyzed reactions. So we have to find the difference, okay, between the prosthetic groups and other coenzymes. And the difference is that the prosthetic groups are tightly bound to the apoenzymes. So option B will be the correct answer. Okay. What is meant by apoenzyme? Apoenzyme is the protein part of the enzyme. Okay. So this is protein portion of the enzyme. It is known as apoenzyme. And prosthetic groups are tightly bound to the apoenzymes, whereas other coenzymes are transiently bound to the apoenzyme. Okay, so the association with apoenzyme is transient. This statement is actually about the coenzymes, not prosthetic groups. Okay, so option B is the correct answer. Question number 64 is from the chapter Cell Cycle and Cell Division. And the question is crossing over takes place between which chromatids and in which stage of the cell cycle. Okay. So first of all, what is crossing over? Let's see. Crossing over. It is the exchange of genetic material between two homologous chromosomes. Okay. Exchange of genetic material between two homologous chromosomes. So it is clear that the chromosomes have to be homologous. So non-homologous will be eliminated here. Okay. So option A and D will be eliminated. Only option B or C can be correct because crossing over takes place between homologous chromosomes. Now, uh, here suppose this is one chromosome, this is another chromosome. Okay, both of them are homologous chromosomes. This is chromosome number 1, this is chromosome number 2. So the chromatids of one chromosome are sister chromatids and the chromatids of two different chromosomes are non-sister chromatids. So the crossing over takes place between the non-sister chromatids like this. Suppose this is first one and this is second one. So the crossing over will take place like this between the non-sister chromatids. Between non-sister chromatids at the Pachytene stage. Okay, Pachytene stage of prophase 1. So the correct answer will be option number B. It takes place between non-sister chromatids of homologous chromosomes at Pachytene stage of prophase 1. So option B is the correct answer. Question number 65 is again related to the chapter cell and the question is Ramachandran plot is used to confirm the structure of options are RNA, protein, triacylglycerides and DNA. So the correct answer is protein. Okay. Ramachandran plot is used to confirm the structure of proteins. It is named after the scientist G.N. Ramachandran. G.N. Ramachandran and it was developed in the year 1963. Okay, and it is used to confirm the structure of proteins. Question number 66 is from the chapters Transport in Plants and the question is which of the following is not a feature of active transport of solutes in plants? First one is occurs against the concentration gradient. This is correct one because active transport takes place against the concentration gradient. Okay. Second one is non-selective. This is not true because this transport is selective. Okay. The membrane proteins are used here if you are aware. Then these membrane proteins are highly selective in nature or you can say highly specific in nature. Okay. Membrane proteins which are known as pumps are highly specific in what they carry across the membrane. Okay. Then third one is occurs through the membranes. It is correct. Okay, that's why membrane proteins are used. Then the last one is requires energy. This is also true. Okay, active transport requires energy because the transport takes place against the concentration gradient. Okay, so the only incorrect one is B, that is non-selective. This is not a feature of active transport. So option B is the correct choice. Now question number 67 is from the chapter Mineral Nutrition, and the question is, which of the following bacteria reduce nitrate in soil into nitrogen? Options are Nitrobacter, Nitrococcus, Thiobacillus and Nitrosomonas. So here question is asked about the conversion of nitrate into nitrogen, that is denitrification. Okay, it is talking about the process of denitrification. And we know that the bacteria Pseudomonas and Thiobacillus are responsible for denitrification. Okay, so Pseudomonas and Thiobacillus. So in the options Thiobacillus is mentioned, so this will be your correct answer. Okay, now regarding other bacteria that are mentioned in the options, let's discuss the biological nitrogen fixation. Biological nitrogen fixation. So what happens during this process? First of all, organic nitrogen is converted into ammonia. Okay, so organic nitrogen gets converted into ammonia, and this process is known as ammonification, isn't it? Then this ammonia is converted into nitrate. Okay, now this takes place in two steps. Okay, ammonia is not directly converted into nitrate. This takes place in two steps. First one is when ammonia is converted into nitrite, and then in the second step this nitrite gets converted into nitrate. Okay, and this is done by different bacteria. So the first one, that is the conversion of ammonia into nitrite, is done by two types of bacteria, nitrosomonas and nitrococcus. Okay, so in the options you can see B and D, these are involved in the conversion of ammonia to nitrite, right? Then the second step, that is the conversion of nitrite into nitrate, is carried out by nitrobacter. Nitrobacter, that is given in the first option. So this is done here. Sorry, this is nitrite to nitrate. So according to this question, our answer is option C, Thiobacillus, because it is asked about the denitrification. Okay. Question number 68 is from the chapter Transport in Plants, and the question is, what will be the direction of flow of water when a plant cell is placed in a hypotonic solution? Options are water will flow in both directions, water will flow out of the cell, water will flow into the cell, and the last one is no flow of water in any direction. So what is a hypotonic solution? Hypotonic solution is nothing but a diluted solution. Okay, diluted solution. And what does that mean? It means the water potential of the solution is higher as compared to that inside the cell. Okay, so let's write it down here. Water potential of the solution is more than the water potential in the cytoplasm of the cell. Okay, so as we know that water will move from the higher potential to the lower potential. Okay, so it will move from the solution into the cytoplasm. Okay, so this will be the movement of water. So the water will enter into the cell, therefore option C will be the correct answer. Okay, and the cell will be said to be turgid. Okay, the cell is known as a turgid cell in this case. And if we talk about the first option, that is water will flow in both direction, this happens in case of isotonic solution. Okay, isotonic solution, and in this case the cell is said to be flaccid. About second option, that water will flow out of the cell, when will it happen? It will happen in case of a hypertonic solution, okay? Hypertonic solution. And in this case, the cell will be said to be plasmolysed. Plasmolysed cell. Okay, so I hope you understood this. Option C will be our correct answer. Question number 69 is from the chapter Respiration in Plants, and the question is, where is respiratory electron transport system, that is ETS, located in plants? Options are mitochondrial matrix, outer mitochondrial membrane, inner mitochondrial membrane, and intermembrane space. So the correct answer is option C, that is inner mitochondrial membrane. Okay. Now what is the electron transport system? It is a metabolic pathway through which the electrons pass from one carrier to another. Okay. So let's write it down here. The electron transport system is the metabolic pathway through which the electron passes from one carrier to another. Right? Now, where is it located? It is present in the inner mitochondrial matrix. It is inner mitochondrial membrane. Mitochondrial membrane. And what is the significance of this system? The significance is that it helps in the release of energy in the form of ATP. Okay. So it helps in releasing and utilizing the energy. So where was this energy stored? It was stored in the NADH plus H plus and FADH2 molecules, right? So energy stored in NADH plus H plus and FADH2 molecules. So this electron transport system helps in releasing this energy from them and also helps in utilizing them. And if you remember that NADH plus H plus was formed during glycolysis, right? It was formed during glycolysis as well as TCA cycle. And FADH2 was formed only during the TCA cycle. Okay. So our answer is option number C, inner mitochondrial membrane. Now question number 70 is from the chapter Photosynthesis in Higher Plants, and the question is, in Hatch and Slack pathway, the primary carbon dioxide acceptor is option A, oxaloacetic acid, option B, phosphoglyceric acid, option C, phosphoenol pyruvate, and option D, rubisco. So the answer is option C, that is phosphoenol pyruvate. Okay. So the primary carbon dioxide acceptor molecule that is asked in the question, it is a 3-carbon molecule. It is a 3-carbon molecule that is known as phosphoenol pyruvate, or in short PEP. PEP. Okay. And where is it present? It is present in the mesophyll cells. Present in the mesophyll cells. And the enzyme responsible for this fixation is PEP carboxylase or PEPcase enzyme. Okay. Enzyme responsible for this fixation is PEPcase. PEPcase. PEP means phosphoenol pyruvate, right? PEPcase or PEP carboxylase. So that is why phosphoenol pyruvate will be our answer here. Now let's move to question number 71. It is from the chapter Plant Growth and Development, and the question is, removal of shoot tips is a very useful technique to boost the production of tea leaves. This is because option A, gibberellins prevent bolting and are inactivated. Option B, auxins prevent leaf dropping at early stages. Option C, effect of auxins is removed and growth of lateral buds is enhanced. And the last option is gibberellins delay senescence of leaves. So the correct answer is option C, that is effects of auxins is removed and growth of lateral buds is enhanced. Okay. So let's see how. First of all, we'll see the roles of the growth hormone auxin. Okay. So the hormone auxin helps to initiate rooting in stem cutting. So it helps to initiate rooting in stem cutting. Then it helps in cell division, elongation, and differentiation. What else? It helps in promoting the flowering. Okay, so it promotes flowering. Then it prevents the fruit and leaf drop at early stages. Prevents fruit and leaf drop at early stages. But at the same time, in matured stages, it promotes the abscission. Okay, so it promotes the abscission of older mature leaves and fruits. Now, here there are several roles of auxin. As you can see in option B also, one of the roles of auxin is mentioned. The auxins prevent leaf dropping at early stages. But according to this question, this will not be the correct answer. Here what we have to remember is that auxin is responsible for apical dominance. Okay. Auxin is responsible for apical dominance. Now, what is apical dominance? See, suppose this is the plant, this is the apical part, right? So, this is apical bud. Now, there are leaves here. So, as we know that the hormone auxin helps in the growth of this apical part, okay? But what happens that growing apical bud inhibits the growth of lateral buds, okay? Growing apical bud inhibits the growth of lateral bud. So this is a problematic situation because we don't want the inhibition of growth of lateral buds. So in order to prevent this, what we do, we just remove the shoot part. Okay. For that, when we do that, what happens? The effect of auxins is removed because the auxin is present in the shoot tips, right? On the tip part, the auxin is available. So when we remove this part, the effect of auxin is removed, and therefore the growth of lateral bud is enhanced because it was inhibited due to the presence of this shoot tip, right? So it is very helpful in the production of tea leaves. Therefore, option C will be the correct answer. Now, question number 72 is from the chapter Photosynthesis in Higher Plants, and the question is, one scientist cultured cladophora in a suspension of azotobacter and illuminated the culture by splitting light through a prism. He observed that bacteria accumulated mainly in the region of, options are violet and green light, indigo and green light, orange and yellow light, and blue and red light. So the correct option is option D, that is blue and red light. Okay. So the scientist that is mentioned in the question was T.W. Angelman. Okay. T.W. Angelman. man. So as mentioned in the question, he used a prism to split the light. Prism to split light into its spectral components. Now, then what did he do? He eliminated green alga, that is cladophora. Eliminated cladophora, which is a green alga, okay? Then he placed the suspension or placed it in the suspension of aerobic bacteria. So that is mentioned in the question. It was azotobacter in a suspension of aerobic bacteria. Now the bacteria was used to detect the sites of oxygen evolution, right? That is why the bacteria was used. Now, what was the significance of this experiment? The significance of this experiment was that the first action spectra of photosynthesis was described. Okay, a first action spectra of, it is a spectrum at least. So a first action spectrum of photosynthesis was described, and it resembled roughly the adsorption spectra of chlorophyll A and B. Chlorophyll A and B. Okay, so the answer will be that the bacteria accumulated in the region of blue and red light of the split spectrum. Okay. Now, question number 73 is from the chapter Plant Growth and Development, and the question is, in order to increase the yield of sugarcane crop, which of the following plant growth regulators should be sprayed? Options are ethylene, auxins, gibberellins, and cytokinins. So the correct option is option C, that is gibberellins. Okay. So one of the important roles of gibberellins is the elongation of the stem. Stem elongation. Okay. So as we know that sugarcane stores carbohydrate as sugar in their stems, right? Now, when this sugarcane is sprayed with gibberellins, what happened? It increases the length of the stem, okay? So due to the elongation of the stem or increase in the length of the stem, the yield is also increased. It increases, or you can say increase in yield takes place by as much as 20 tons per acre. Therefore, gibberellins are widely used in case of sugarcane. Question number 74 is from the chapter Sexual Reproduction in Flowering Plants. And the question is, what type of pollination takes place in Vallisneria? Option A, pollination occurs in submerged condition by water. Option B, flowers emerge above the surface of water and pollination occurs by insects. Option C, flowers emerge above water surface and pollen is carried by wind. And the last option is male flowers are carried by water currents to female flowers at the surface of water. So the correct answer will be option D. Okay. Male flowers are carried by water currents to female flowers at the surface of water. Okay. So first of all, this Vallisneria, it grows in fresh water. What kind of water? Fresh water. Okay. Now what happens, how the pollination takes place. The female reaches the surface by the long stalk. Okay, so if this is the surface of the water, then female is present inside the water, right? This is the female plant. So this will reach the surface of the water by long stalk. Reach the surface by long stalk. So we are talking about the female flower right now. Now, what happens to the male? The male flower, or you can say the pollens, are released on the surface. So these are the pollens. These are released on the surface. Released on surface, water surface. And then these are carried by the water currents towards the female part. Okay. Carried by water currents towards female flower, or you can say it reaches the stigma by the water currents. Therefore, option D will be the correct answer. Next question is again from the chapter Sexual Reproduction in Flowering Plants. And the question is, in which one of the following both autogamy and G-tonogamy are prevented? Options are wheat, papaya, castor and maize. So the correct answer is option B, that is papaya. Okay. What is autogamy? Autogamy is a kind of pollination in which the pollen is transferred from the anther of one flower to the stigma of the same flower. Okay. So pollen is transferred to the stigma of the same flower. Okay. Same flower. And what happens in case of G-tonogamy? Here the pollen from the anther of one flower is transferred to the stigma of another flower. Stigma of another flower. But both the flowers are on the same tree or the same plant. Okay. So here the flowers are different, but the plant is still the same. So genetically both are same. Now there is another type of pollination known as xenogamy. Xenogamy. Here the pollen is transferred from the anther of one flower to the stigma of another flower that is present on another tree, okay, or another plant. So pollen is transferred to the stigma of flower present on different plant. So option B is correct because papaya exhibits xenogamy. Okay, example papaya, and the reason is that papaya is a dioecious plant. Dioecious, or you can say unisexual plant. So here the males and females are found on different plants. Male and female flowers are present on different plants. Therefore, only xenogamy is possible. So autogamy and G-tonogamy both are prevented in this case. Therefore, option B is the correct answer. Now question number 76. It is from the chapter Morphology of Flowering Plants. And here we have to match the placental types that are mentioned in column 1 with their examples mentioned in column 2. Okay. So the first type of placenta that is mentioned is basal. So what happens in case of basal placentation? Here the placenta develops at the base of the ovary. Okay. The placenta develops at the base of the ovary. The ovary, and a single ovule is attached to it. Okay. And single ovule is attached to it. Examples of basal placentation will be sunflower and marigold. So the correct match for A will be option number 4, that is sunflower. Okay. So the correct match for A will be 4. Then we come to the second one, that is axile placentation, right? Axile. So as the word suggests, it is related to axis. So here the placenta is axial. So the placenta is present on the axis. So it is axial. Therefore, it is known as axile placentation. And here there are more than one ovules that are attached to it in multilocular ovary. Okay. And ovules attached to it in a multilocular ovary. And what will be the examples of exile placentation? Examples are China rose, then tomato and lemon as well. So here China rose is mentioned, therefore it will be matched with China rose. Okay, so the correct match for B will be number 2. Now the third one is parietal. Parietal placentation. So what happens in case of parietal placentation? Here the ovules are developed on the inner wall of the ovary. Okay, the ovules are developed on the inner wall of the ovary, or you can say peripheral wall. Okay, peripheral part of the ovary. So the examples of parietal placentation is mustard and argemone. Mustard and argemone. So which of them is mentioned here? Here mustard is mentioned, so the correct match for parietal will be number 1, that is mustard. Okay, C will be matched with 1. Now the last one is free central. Free central placentation. So here the ovules are borne on the central axis. Okay, ovules borne on the central axis. Therefore, it is known as free central. And here there is no septa. Okay, septa are absent. Septa absent. In case of the parietal, there was a septa. There was a false septa. Okay, false septa present. And due to that false septa, the ovary which was actually one chambered had become two chambered. Okay, but in case of free central, there are no septa. And the examples of free central will be dianthus and primrose. Dianthus and primrose. So which of them is mentioned here? Dianthus is mentioned, so the correct match for D will be 3. So out of the given options, which are the correct matches? It is option number C, right? So option number C will be the correct answer. Now question number 77 is from the chapter Biotechnology, Principles and Processes. And the question is, a selectable marker is used to, option A, help in eliminating the non-transformants so that the transformants can be regenerated. Option B, identify the gene for a desired trait in an alien organism. Option C, select a suitable vector for transformation in a specific crop. And the last option is mark a gene on a chromosome for isolation using restriction enzyme. So the correct answer is option A, that the selectable marker helps in eliminating the non-transformants so that the transformants can be regenerated. Okay. And what are the transformants? Transformants are the cells that contain foreign genetic material. Cells that contain foreign genetic material. Okay. So since transformants contain the foreign genetic material, so we need more number of transformants so that more genetic materials can be transferred, right? Genetic materials are the desired genes that we want to insert in the organism. Okay. So what does selectable marker do? It first identifies and eliminates the non-transformants. Let's write it down here. Identifies and eliminates the non-transformants and then it selectively permits the growth of transformants. Selectively permits the growth of transformants. And the useful selectable markers for E. coli are what? The examples are the genes encoding resistance to antibiotics. Okay, genes encoding resistance to antibiotics, such as ampicillin or chloramphenicol, etc., are considered as good selectable markers for E. coli. Okay, so option A is the correct answer. Question number 78 is from the chapter Biodiversity and Conservation, and the question is Western Ghats have a large number of plant and animal species that are not found anywhere else. Which of the following terms will you use to notify such species? Options are endemic, vulnerable, threatened, and keystone. And the correct answer is option A, that is endemic. Okay, so this is related to the term endemism. Endemism, it means that the species are confined to that region only. Okay, species confined to a particular region and they are not found anywhere else. Not found anywhere else. So the example mentioned in the question is used for endemic species. Okay. Question number 79 is from the chapter Environmental Issues, and the question is which of the following statements about ozone is correct? Okay, so the first statement says tropospheric ozone protects us from UV radiations. This is not true. Okay, see, we know that troposphere is the lower atmosphere, right? This is the troposphere, and above the troposphere we find stratosphere, right? Stratosphere. And in both these layers, troposphere as well as stratosphere, ozone is found, but ozone in the troposphere is known as bad ozone. Okay, it is considered as bad ozone, whereas the ozone in the stratosphere is considered as good ozone because it protects us from the harmful UV radiation. So first statement says that tropospheric ozone protects us from UV radiation, which is not true because tropospheric ozone is a bad ozone. Okay. Second statement says stratospheric ozone is bad. This is not true because tropospheric ozone is bad. Third statement says tropospheric ozone is good. This is also not true. The case is just the opposite. And the last statement says stratospheric ozone protects us from UV radiation. This is correct. Okay, therefore stratospheric ozone is known as good ozone. Therefore option D is the correct answer. Question number 80 is from the chapter Biodiversity and Conservation, and the question is exploration of molecular, genetic and species level diversity for novel products of economic importance is known as, options are biopiracy, bioenergetics, bioremediation and bioprospecting. And the correct answer is option D, that is bioprospecting. Okay, so this is a direct question. You just have to remember the definition of bioprospecting. Okay, it means the exploration of molecular, genetic and species level diversity for novel products of economic importance. Question number 81 is from the chapter Environmental Issues, and the question is which of the following is an innovative remedy for plastic waste? Options are burning in the absence of oxygen, then burying 500-meter deep below soil surface, then polyblend, and the last one is electrostatic precipitator. So the correct answer is option C, that is a polyblend. So this is very innovative remedy for plastic waste. And what is polyblend? It is nothing but fine powder of recycled modified plastic. Okay, fine powder of recycled modified plastic. Okay, it was invented by Ahmad Khan. invented by Ahmad Khan. This is mentioned in your NCERT in the year 1998. And the raw material for polyblend is any plastic film waste. Okay, raw material can be any plastic film waste. So I hope you understood this. Alright, now question number 82 is from the chapter Organisms and Population, and the question is, between which among the following the relationship is not an example of commensalism? So what is commensalism? It is the relationship between the organisms where one species is benefited, one species is benefited and the other is neither benefited nor harmed. Okay, other neither benefited nor harmed. Now let's see what are the options here. First option is orchid and the tree on which it grows. So we know that orchid grows as an epiphyte on the mango tree. So orchid is benefited, but the mango tree derives no benefit from it. So obviously this is the example of commensalism. Commensalism. Now, second one is cattle egret and grazing cattle. Again, this is the example of commensalism because grazing cattle, when they move, they just flush out the insects that were earlier hidden from the egret. Okay, so now the egret could easily catch those insects. Okay, so this way grazing cattle help the cattle egret. So cattle egret are benefited, but the grazing cattle do not derive any benefit. So this is the example of commensalism again. Third one is sea anemone and clownfish. Again, this is the example of commensalism. Okay, so we know that the sea anemone have stinging tentacles, stinging tentacles, and the clownfish remain close to the sea anemone. So what happens? Due to the stinging tentacles, the predators of the clownfish do not come close to them. Therefore, these are protected from their predators. Okay, so these protect the fish from their predators. But does sea anemone derive any benefit? No, it has no benefit nor any harm. So this is also the example of commensalism. The last one is female wasp and fig species. This is not the example of commensalism. This is the example of mutualism. Okay, mutualism, that means both derive the benefit. Okay, both the organisms or both the species derive benefit from each other. So what happens? Female wasp, it pollinates the fig species. Female wasp, it pollinates the fig species while searching for a safe place to lay eggs, right? And what does fig do to female wasp? In return, it offers some growing seeds. Okay, offers growing or developing seeds for the developing larva of the wasp, right? Developing wasp larva. This way, this is a mutual relationship. This is mutual relationship. So this is not the example of commensalism. So option D is the correct answer. Now question number 83 is from the chapter Environmental Issues, and the question is, if an agricultural field is liberally irrigated for a prolonged period of time, it is likely to face problem of, options are metal toxicity, alkalinity, acidity and salinity, and the correct option is option D, that is salinity. Okay, so we know that nowadays improper resource utilization is also leading to problematic situation, right? And waterlogging and soil salinity is one of them. So what happens when irrigation is done without proper drainage of water? It leads to waterlogging. Okay, so let's write it down here. Irrigation without proper drainage of water. So water is coming, but there is no way for water to move out from that place. So it leads to a situation that is known as water logging. Okay, water is collected in that place. Water logging in the soil. So it definitely affects the crops, but other than that, it also draws salt to the surface of the soil. Okay, draws salt to the surface of the soil. Then this salt is deposited as a thin crust on the land surface. Salt is deposited as a thin crust on land surface, and it starts collecting at the roots also, and starts collecting at the roots of the plant. This way it increases the soil salinity, right? Therefore option D will be the correct answer. Question number 84 is from the chapter Microbes in Human Welfare, and the question is, which of the following statements about methanogens is not correct? So what are methanogens? These are the bacteria that produce methane gas, right? Bacteria that produce methane gas. Now, the first option is they can be used to produce biogas. This is a correct statement. Then they are found in the rumen of cattle and their excreta. Rumen is a part of their stomach. So this is also a correct statement. Then they grow aerobically and break down cellulose rich food. So this is, see, the aerobically term is not correct because they grow anaerobically. Okay. Anaerobically. And the second part is correct, that they break down the cellulose rich food that is present in the stomach of cattle. Okay, and this way it also provides nutrition to the cattle. Then the last one is they produce methane gas. This is correct statement. So the only incorrect statement that we had to find is option number C. Okay, so C is the correct choice. Now next question is from the chapter Strategies for Enhancement in Food Production, and the question is, in mung bean resistance to yellow mosaic virus and powdery mildew were brought about by, options are mutation breeding, biofortification, tissue culture and hybridization and selection, and the correct answer is option A, that is mutation breeding. So what is the meaning of the word mutation? The term mutation is used for the process by which the genetic variation are created, right? So the variation in genes are done, okay, through the changes in the base sequence within the genes. And what does it result in? It results in the development of new trait or new character. New trait or character. So this is what is exactly done here in case of mutation breeding. Okay, here in case of mutation breeding, there is inducing the mutation. The process of it is the process of inducing the mutation artificially. Got it? So mutation breeding, what is done here? It is the inducing of mutation mutation artificially through the use of chemical or radiation. And then what is done? Selection is done. Okay, then the process involves selecting and using the plants with desirable character, desirable character as the source of breeding. Got it? So first mutation is brought about, okay? That is, mutation is induced artificially, and then the plants which have the desirable character are selected. Got it? So therefore option A will be the correct answer. Question number 86 is from the chapter Human Health and Diseases, and the question is coca alkaloid or cocaine is obtained from. Options are Papaver somniferum, Atropha belladonna, Erythroxylum coca, and Datura. And the correct answer is option C, that is Erythroxylum coca. Okay. So what is cocaine? It is commonly called coke or crack. Okay. So the common name of cocaine is coke or crack. And how is it consumed? It is usually snorted. Okay. So what happens when it is snorted? That means what are the effects of cocaine? Let's list them here. Effects of cocaine. So it basically interferes with the transport of a neurotransmitter. Okay. Which neurotransmitter? Dopamine. So it interferes with the transport of neurotransmitter dopamine. So what kind of effect it produces? It has a potent stimulating action on central nervous system. Okay. Potent stimulating action on the central nervous system. Okay. This is action, due to which a sense of euphoria is produced. So it produces a sense of euphoria. Euphoria, or you can say increased energy. So the person feels very energetic after consuming cocaine. But obviously there will be side effects. So excessive dosage of cocaine can lead to hallucination. Okay, hallucination. So this was all about this. So as we can see that coca alkaloid or cocaine is obtained from the plant that is known as Erythroxylum coca, and this plant is native to which country? It is native to South America. Okay. So option C is the correct answer. Question number 87 is from the chapter Microbes in Human Welfare, and the question is among the following pairs of microbes, which pair has both the microbes that can be used as biofertilizers? Okay, so what are biofertilizers? These are the organisms that are used as fertilizers, right? So the definition will be that biofertilizers are the organisms that enrich the quality of the soil. Okay, because that's what fertilizer do. Quality of the soil. And the main source of biofertilizers, main source are bacteria, then fungi and cyanobacteria. Okay, so these are most widely used biofertilizers. Now here the options are, first one is Aspergillus and Rhizopus, second one is Rhizobium and Rhizopus, third one is Cyanobacteria and Rhizobium, and the last is Aspergillus and Cyanobacteria. So we have already seen that one of the important sources is, the main source is Cyanobacteria. So obviously Cyanobacteria is there in the third option, and then Rhizobium, that is a bacteria. So this is also an important source of biofertilizers. So option C will be the correct answer. It is actually the biofertilizer. Okay, it is not the source, it is actually a biofertilizer. So what is the significance of rhizobium? Do you remember? It forms symbiotic association with the roots of leguminous plants, right? So let's write it down here. Rhizobium, this is an important point, that rhizobium forms symbiotic association with the roots of leguminous plants. Okay, leguminous plants. So they form nodules at the roots of leguminous plants, right? And most important the function of rhizobium is that they fix the atmospheric nitrogen into organic form. Okay, fix atmospheric nitrogen in organic form, and this is used by the plants as nutrients. Okay, form that is used as nutrient by the plant. So our answer is option number C. Now, question number 88 is from the chapter Biotechnology, Principles and Processes. And the question is, given below are four statements pertaining to separation of DNA fragments using gel electrophoresis. Identify the incorrect statement. Okay. So it is already mentioned in the question that gel electrophoresis is being used to separate the DNA fragments, right? Then we have to find out the incorrect statement. Now, let's see what are the statements mentioned here. First statement says DNA is negatively charged molecule and so it is loaded on gel towards the anode terminal. This is incorrect statement. Why? Because although DNA is negatively charged molecule, it is not loaded towards the anode terminal, it is loaded on another side and it is forced to move towards anode. Okay, it is forced to move towards the anode. It is not loaded towards the anode. Therefore, this is an incorrect statement. Then the second statement says DNA fragments travel along the surface of the gel whose concentration does not affect movement of DNA. Again, this is incorrect. Why? Because see, the concentration of the gel actually affect the movement of DNA. And the reason is that the agarose gel, let's write it down here. The agarose gel that is used in this case as the medium or matrix provides the sieving effect. Okay, it provides the sieving effect to the movement of the DNA fragments. Now, sieving effect is determined by the size of the pores. Okay. Now, what happens when the concentration of the agarose is increased? The concentration of agarose is more, then what happens? The pore size decreases. Pore size decreases, and as we have seen that the size of pores affect the sieving effect, therefore the concentration of agarose will also indirectly affect the sieving effect. Okay, so this is the incorrect statement again. Now we come to the third statement. It says smaller the size of DNA fragment, larger is the distance it travels through it. This is a correct statement. Okay, so the smaller sized fragment will move farther. Now, the last one is that pure DNA can be visualized directly by exposing UV radiation. Again, this is an incorrect statement because we know that before exposing UV radiation to the DNA fragment that is obtained here, we have to use the ethidium bromide, right? Ethidium bromide. So, the pure DNA has to be stained with the compound known as ethidium bromide, and then it has to be exposed to UV radiation, not directly to UV radiation. So we can see that only the third statement, that is statement C, is correct and all others are incorrect. So A, B and D are incorrect. So option D will be the correct answer. Question number 89 is again from the chapter Biotechnology Principles and Processes. And the question is, an enzyme catalyzing the removal of nucleotides from ends of DNA is, options are DNA ligase, endonuclease, exonuclease and protease. So the answer will be option C, that is exonuclease. Okay. So we know that the larger class of enzyme that is known as nuclease helps in the removal of nucleotides. Now there are two types of nucleases. First one is exonuclease and the other one is endonuclease. So what is the difference between them? As the word suggests, exo and endo. Exo means outer, endo means inner. Okay, it is related to outer and inner. So exonuclease, it removes the nucleotides from the ends of the DNA. Okay, removes the nucleotides from the ends of DNA. And this is asked in the question. Whereas the endonuclease make cuts at specific position within the DNA. They make cuts at specific position within the DNA. The first one is DNA ligase. What is DNA ligase? It is actually the glue type, it acts as a glue. It acts as glue and it joins the DNA fragments. Joins the DNA fragments. So the nuclease, what they do nuclease remove the DNA fragment and DNA ligase, they join the DNA fragments, right? The last one is protease. Protease is very clear. It helps in the breakdown of protein. Breakdown of proteins. So I hope all the terms are clear to you. The answer according to the question will be option C, that is exonuclease. And now the last question is from the chapter Biotechnology and its applications. And the question is, in RNA interference, I for interference, the genes are silenced using. Options are double stranded RNA, single stranded DNA, single stranded RNA and double stranded DNA. And the correct answer is option A, that is double stranded RNA. Okay. So if you are aware, the RNA interference takes place in all the eukaryotes. Okay. It takes place in all eukaryotes. And what is the significance of RNA interference? It is used as a method of cellular defense. Okay, as a method of cellular defense. Now, how does it do so? See, first of all, a complementary double-stranded RNA will bind to a specific mRNA. Okay, a double-stranded, a complementary double-stranded RNA binds to the specific mRNA. So what happens? Due to this, the mRNA is silenced. mRNA gets silenced. That is, double-stranded RNA is involved in silencing of this mRNA. Okay. Therefore, option A will be the correct answer. So here we complete all the questions from NEET Odisha 2019 biology paper. I hope things were clear to you. To have a better understanding of the questions, I would suggest you to practice the questions from our website www.examsnet.com. For more information, download our app from the link given in the description. Okay. Till then, keep learning, keep scoring high. We'll get back with another session soon. Thank you so much.